Hello everyone and welcome to today's webinar, Domoic acid, research on the effects of repeat low-level exposures and its implications for human toxicity. Before we get started, I'd like to go over a few items so you know how to participate in today's event. You may join the webinar using your computer speaker system as the audio, which is the default. If you prefer to join over the telephone, just select telephone in the audio pane and the dial-in information will be displayed. For technical difficulties, please use the questions box of the control panel. You can submit questions to today's presenters by typing your questions into the questions box of the control panel. You may send in your questions at any time during the event. We will collect these and address them during the brief Q&A sessions following each presentation or during the panel discussion at the end of today's session. We have included handouts for today's session. You can download them from the handouts panel. Today's webinar is being recorded. I would now like to introduce Lauren Zeiss, Director of the Office of Environmental Health Hazard Assessment, or OEHA. Lauren?
Thank you, Tracy. On behalf of OEHA, I'd like to welcome everyone to today's webinar on domoic acid. OEHA is one of 6 departments of the California Environmental Protection Agency. Our mission is to protect human health and environmental health through the scientific evaluation of risks. We're the lead agency for assessment of health risks posed by environmental contaminants, including domoic acid in seafood. OEHA reviews data on domoic acid levels in seafood, and when they're too high, in consultation with our state public health department, we recommend to the California Department of Fish and Wildlife closures of the affected fisheries. We also make recommendations to reopen fisheries when levels have cleared. Today's webinar builds on the 2017 workshop we held with the University of California, Davis on the state of the science of the health effects of domoic acid. Today we are updating our understanding of domoic acid exposure by examining the very recent science on neurological and developmental effects associated with repeat exposure to low levels of domoic acid. As we listen to the presentations, we'll keep in mind a couple of points. First, how the recent findings from non-human primate studies and human studies may shape our understanding of vulnerable populations, what their potential risks for domoic acid toxicity may be, the types of neurotoxicity we might be concerned about, as well as levels of concern. And second, what the research might suggest about the vulnerability of high-frequency shellfish consumers who may be repeatedly exposed to low levels of domoic acid. So with that, thank you everyone for joining us. I'd like to now introduce and turn the webinar over to our webinar organizer, Dr. Shannon Murphy. Shannon is a staff toxicologist in OEHA's Fish, Ecotoxicology, and Water section. Shannon?
Thanks, Lauren. Let me add my welcome. Today's speaker session will begin with 2 short presentations by OEHA. The first is a primer on how the current federal action levels for domoic acid were derived, followed by a recap of the OEHA UC Davis State of the Science Domoic Acid Workshop of 2017. Just a reminder that the brief question and answer period following each presentation is intended for short, clarifying questions. More in-depth questions may be addressed in the extended panel discussion session. Our first speaker is Dr. Susan Claising, Chief of the Fish, Ecotoxicology, and Water section at OEHA. Welcome, Susan.
Thank you, and I'd like to welcome everyone again for joining us today. As noted, I'm going to be giving just a brief explanation of how the action levels for domoic acid were derived. First, what are the action levels for domoic acid? Just to quote FDA, action levels and tolerances represent limits at or above which FDA will take legal action to remove products from the market. My office and the California Department of Public Health use FDA domoic acid action levels to make closure, delay of opening, and reopening recommendations to the California Department of Fish and Wildlife. Those action levels are 20 parts per million in all fish, which include finfish, crustaceans, and mollusks, except the action level is greater than 30 parts per million in Dungeness crab viscera. Our authority to make such recommendations come under Fish and Game Code Section 5523. Just a little background on how we first learned about domoic acid toxicity in humans. There was a poisoning incident that originated in Prince Edward Island, Canada in 1987. Initially, 3 patients presented with rapid onset of disorientation, confusion, and memory loss over a few-day period. All of these individuals had eaten cultured mussels that originated from Prince Edward Island. At that time, domoic acid was not known to be a marine biotoxin, and so it wasn't on the radar when they were looking for a causative agent. These exposures were acute, most likely a single meal in many cases. There were more than 250 reports of illness associated with consumption of mussels. 107 people met the case definition. 19 people were hospitalized, 16 treated in the ICU, 3 died while hospitalized, and a 4th severely ill patient died of a heart attack a few months later. That person is often included in the mortality statistics for this event. Symptoms began within 15 minutes to 38 hours after consumption. There were 2 types of symptoms. Milder symptoms were gastrointestinal in nature. They generally began within 24 hours. Nausea, vomiting, cramps, and diarrhea. The more severe symptoms were neurological in nature, seizures, coma, and irreversible memory loss. They typically began within 48 hours. It's the irreversible memory loss that led to the moniker for this disorder of amnesic shellfish poisoning, or ASP. They determined that the most sensitive populations during this outbreak were older, tended to be men, and those with pre-existing medical conditions such as diabetes or kidney disease. Just a little background on risk assessment for those of you who don't do it for a living. One of the things that we're looking for is a dose response curve, the dose plotted against whatever adverse outcome that we are concerned about. A typical classic dose response curve looks something like this. And 2 key things that we're looking for are referred to as points of departure. The first is a NOEL, or no observed adverse effect level, and that is the highest dose that is not significantly different from the control. The control would not have been exposed. Another is the LOEL, or lowest observed adverse effect level, and that is the lowest dose that is significantly different from the control. So there may be other doses, higher, lower, intermediate, but those are 2 key points that we're looking for. It's important to know that these values are based on the experimental data that we have, whatever doses there were. The true values for the NOEL and LOEL for the population are likely unknown. So the risk assessment that was done for domoic acid after the Prince Edward Island event, there was a dose response estimation that was based on data that they had for 10 individuals for whom they had a sample of uneaten mussels. Those individuals ranged in age from 60 to 84 years. It was 9 patients plus 1 subject who did eat the mussels but did not get sick. 3 of the patients were hospitalized. They collected uneaten mussels from these individuals and found them to contain between 310 and 1,280 parts per million domoic acid. The estimated individual consumption rates ranged from 35 to 400 grams of mussels. When they combine these values, the individual estimated domoic acid consumed ranged from 20 milligrams in the person who did not get sick to 290 milligrams in 2 individuals who were the sickest. So this table is adapted from Pearl et al., who published a paper in the New England Journal of Medicine in 1990 and several other papers as well. And you can see that this shows the estimated amount of domoic acid consumed in the second column, rated from the lowest to the highest. So 20 milligrams in the person who did not get sick, and 60 milligrams was the lowest dose that showed any symptoms. In the 4 right columns, you'll see the 4 types of adverse effects that they were looking at. GI effects, memory loss, were they hospitalized, and were they in the ICU? And you can see a clear positive dose response here. The higher the amount of DA that people were exposed to, the more likely they are to have 1 or more of these adverse effects. So we plotted that data in a way similar to a dose response curve, with the dose along the x-axis and the type and number of effects the 0, 1, 2, 3, or 4 on the y-axis, and you'll see that it plotted to be somewhat similar to the classic dose-response curve that I showed you a few slides ago, except there was one individual that had more severe effects than would be predicted based on a typical dose-response curve. So I'm going to show you now how the domoic acid action levels were derived. And if you look in the literature, you'll find that there are various ways of deriving the number. Different agencies in different countries, and they use different body weights, different consumption rates, different uncertainty factors, but they all came up to the same number. So I'm going to show you the way that FDA has explained this in some of their documents. First, 240 parts per million was determined to be the lowest effect concentration in muscle meat, and that's the 60 milligram lowest dose cause and effect divided by an estimated 0.25 kilo serving size. This was divided by an uncertainty factor of 10, leading to 24. Just as an aside, uncertainty factors are always part of a risk assessment and are used to account for data deficiencies, such as inter-individual differences, small sample size, lack of chronic exposure data, and using a low L versus a no L in the calculation. Uncertainty factors for human data typically range from 10 to 300. So that led to an action level of 20 parts per million in seafood. Additionally, a tolerable intake was derived, taking that same 60 milligram lowest effect dose divided by the same 10-fold uncertainty factor, leading to a tolerable intake of 6 milligrams of domoic acid. So Health Canada originally derived the action level, and it was adopted by FDA. And then subsequent to that, it was determined that there should be a separate action level for Dungeness crab viscera. Viscera tends to accumulate more domoic acid, and there's less of it in a crab. So data were submitted to FDA, and FDA made several assumptions. One is that a person eats one crab, including the viscera, in an eating occasion. That 300 grams is a reasonable estimate of crab meat yield from one crab, and that 6 ppm would be a potential concentration of DA in crab meat. So using that 6 ppm with the 0.3 kilo, they estimated that there would be approximately 1 milligram of DA contribution from the crab meat. And you'll see that there's rounding in all of these equations. They further estimated that there would be 150 grams as a reasonable estimate of crab viscera from one weight. So if 1 milligram would be an estimate of contribution from the crab meat, and a tolerable intake is 6 milligrams, then that would allow 5 milligrams to be the contribution from crab viscera. So that with that 5 milligrams being the tolerable amount of domoic acid and 150 grams of crab viscera consumed in one eating occasion, that came out to 33, which was rounded to 30 parts per million as the tolerable amount of DA in crab viscera, and the action level became greater than 30 parts per million in crab viscera. So why are we all here today? We've had recent domoic acid events in California. In 2015-16, we had an unprecedented Pseudonychia bloom that affected the U.S. Pacific coast. Certain strains of Pseudonychia produced domoic acid under certain conditions. At that time, the California Dungeness crab fishery opening was delayed up to 5 months in parts of the state. It resulted in a fishery closure resource disaster and a commercial fishery failure declaration for both the Dungeness and rock crab fisheries for the 2015-16 season. We've had closures and delay of openings occurring during subsequent crab and lobster seasons, and our recreational razor clam fishery has been closed in Humboldt and Del Norte counties since April of 2016. Subsequently, there have been publications and workshops related to domoic acid. In 2016, the California Ocean Science Trust published 2 documents, an FAQ on harmful algal blooms in California fisheries, another document framing the scientific opportunities on harmful algal blooms in California fisheries. In 2017, it's already been noted that OEHA and UC Davis co-sponsored a 1-day workshop to evaluate the state of science on domoic acid and the implications for human toxicity. And in 2018, the California Ocean Protection Council and California Ocean Science Trust co-hosted a HAB workshop on how the scientific understanding of HABs can inform domoic acid monitoring for seafood safety. And that's the end of my talk.
Thank you, Susan. We have a couple general questions from the audience. Some of you have asked if the PowerPoint presentations will be available for sharing. What I would recommend is you go ahead and email the organizer, Shannon Murphy, myself at shannon.murphy@oeha.ca.gov, and I will put you in touch with the authors of each presentation, and you can contact them directly to make that arrangement if they so choose. Also, too, we are recording this webinar, and we intend to post it at a future date, and we're exploring the technical factors associated with that, so please stay tuned. We do have another question. This is for you, Dr. Claesing. How was DA, or domoic acid, identified as the active agent in the first ASP event?
There's a really excellent article on it, which I have not reread recently, so I can't give you the details, but I can say that there was a very concerted effort that went actually very quickly on how they were able to rule out other potential causative agents and focus in on finding that it was domoic acid.
We have a second question here. Can you explain the relationship between pseudonychia and domoic acid?
Well, pseudonychia is a marine diatom that under certain conditions and certain species will produce domoic acid.
Thank you very much. Not seeing any more questions, we'll go ahead and move forward. Just a reminder that attendees can submit questions at any time during the webinar through the question pane of the control panel. Also, another note is that we have handouts available, and if you look at the control panel under handouts, you'll be able to download those there. In this next presentation, I will provide an overview summary of our 2017 domoic acid workshop. A key aim of today's presentations and discussion is to build upon the 2017 State of the Science workshop. A copy of the 2017 workshop agenda, including a list of all the featured presenters, is available on OEHA's website. Before we dive into the new science, let us revisit some of the historical domoic acid highlights. 2017 marked the 30th anniversary of the 1987 Prince Edward Island domoic acid event. That public health episode spurred teams of scientists, health agencies, and policymakers to identify, understand, and manage this mystery agent we now know as domoic acid. Health symposia and workshops directly followed, with the 20 ppm action level for domoic acid in seafood adopted by the FDA in 1992, and later by the European Union in 1997. During the next 2 decades, the Pacific Coast witnessed sporadic episodes of bird deaths and marine mammal illness and strandings attributed to domoic acid toxicity. These events were a prelude to the massive harmful algal bloom event of 2015-2016, where high levels of domoic acid impacted fisheries along the whole Pacific Coast, closing the entire California coast to Dungeness crab fishing for most of the fishing season. This season of The Blob launched a new series of workshops and public meetings, thrusting scientists, health officials, and marine resource managers and the fishing community into direct confrontation with domoic acid, a challenge that continues with our discussion today. Among these events was the 2017 State of the Science workshop hosted by OEHA in collaboration with the University of California, Davis, which featured a broad spectrum of presenters from experts in domoic acid toxicity. Here are the highlights of that conference. Dr. Tasker of Prince Edward Island University, a researcher and ground zero witness to the seminal domoic acid event, opened the day with a first-hand recounting of November 1987. Applauding the valiant efforts of those scientists and health officials, Tasker outlined how these individuals worked in record time to identify the cause and minimize the impact of the crisis. The early years shed light on domoic acid toxicity with the naming of the syndrome, defining of the symptomology, establishment of the action level, and further description of domoic acid's impact beyond the acute effects, setting the stage for a more complex profile of neurotoxicity. Dr. Golan of the Marine Mammal Center of Sausalito described the effects of domoic acid toxicity on marine mammals, particularly sea lions. Called sentinels of the sea, California sea lions exhibiting signs of DA intoxication can indicate the presence of a recent or active bloom and the potential health implications for human exposure. Dr. Golan described both the acute and chronic neurotoxic syndromes, highlighting that the fetal compartment, especially the amniotic fluid, acts as a sink or storage depot for domoic acid accumulation. These findings suggest that the fetus may represent a vulnerable population. From the sea to the shore, Dr. Gratton of the University of Maryland School of Medicine examined the impacts of low-level DA exposure in humans from consumption of razor clams through her efforts with the coastal cohort study involving tribes of the Pacific Northwest. Neurobehavioral assessments and neurocognitive scoring suggest that these low-level exposures are associated with memory decline in high-frequency razor clam consumers. We will welcome Dr. Gratton later in the webinar for an update on this research. Dr. Ramsdale of NOAA delivered a primer on the interesting pharmacokinetics of DA, noting that this glutamine receptor compound acts as a partial agonist with high affinity for kainate receptors. DA induces neuroexcitotoxic effects, in part by failing to effectively desensitize the glutamate receptor. DA's kinetic profile of poor absorption and rapid elimination make it a challenging compound to model. Age, the presence of kidney disease, and pregnancy complicate one's susceptibility to DA's toxic effects. Dr. Isoharinen's pharmacokinetic work in the context of pregnancy will further this discussion later in the webinar. Dr. Faustman of the University of Washington expanded the discussion about low-level DA exposure during various developmental stages, critically examining the spectrum of neurotoxic effects associated with in utero exposures in mice. Altered motor coordination and changes in startle response observed in the offspring of dams exposed during pregnancy, coupled with changes in the kinetic profiles of pregnant versus non-pregnant adults, and concentrated levels of domoic acid in the amniotic fluid, model effects similar to those observed in the offspring of DA-intoxicated marine mammals. The work of Dr. Doucet of the University of Prince Edward Island further advanced the conversation about early life exposure and adverse neurodevelopmental effects, noting that rat pups exposed during early postnatal life demonstrate a varied spectrum of disrupted or delayed neurocognitive development. Depending on the duration and the timing of exposure, aberrant behaviors indicative of changes in learning and memory may remain latent for a time, manifesting later in life during another key window of development. We will revisit this concept of early DA exposure and the implications for neurocognitive development during Dr. Burbacher's presentation today. Finally, Dr. Lavis of the Oregon Health Sciences University fostered a stimulating discourse on the similarities between DA-induced neurotoxic effects observed in rodent models and marine mammals, with elements of the neurobehavioral profile observed in children with autism spectrum disorders. Altered brain connectivity and aberrant social behaviors observed in exposed rodents, and disruptions in highly coordinated social behaviors and increased aggression noted in intoxicated sea lions mirror behaviors often noticed in children with these autism spectrum disorders. As we prepare for today's discussion, we note that with this increased frequency and duration of these harmful algal bloom events, the intoxication syndromes observed in marine mammals, the neurodevelopmental implications from decades of work in laboratory models, and the emerging associations from social science studies, that collectively, the 2017 State of the Science Workshop highlighted the need for further examination of low-level domoic acid exposure, the nuanced spectrum of neurotoxic effects, and their nexus with identification of potentially vulnerable populations, hence the focus of today's webinar. And with that, we'll have a look at any of your questions. Okay. It looks like we just have a quick question about the handouts, and it looks like our organizer has addressed that. But once again, we have a couple handouts for people to look at, including the agenda and bio sketches of the speakers. All right. So moving ahead, I would like now to introduce Dr. Thomas Burbacher, who is going to discuss maternal and offspring effects of chronic low-level domoic acid exposure in non-human primates. Dr. Burbacher is a professor of environmental and occupational health sciences, division head of reproductive and developmental science, and director of the Infant Primate Research Laboratory at the University of Washington's National Primate Research Center in Seattle. His research investigates changes in brain development and function caused by perinatal exposure to neuroactive substances and the adverse human health implications, including developmental disabilities related to exposure to environmental contaminants. Dr. Burbacher is joined by PhD student Rebecca Petroff, whose efforts contribute to research exploring the neurological effects of low-level chronic domoic acid exposure using behavioral observations and MRI studies in the non-human model. Welcome, Ms. Petroff and Dr. Burbacher.
Thank you. Thank you very much. Welcome, everyone. I'm going to begin by giving you an outline of what we're going to talk about this morning. Want to make sure that we talk about the study goals. What were the main reasons that we initiated this study? Talk a little bit about the non-human primate model, and then get into our original study design and procedures that we used, and go over the results, the published results for those that we have so far. And then we're going to talk about a bit of additional research that we were able to do with a supplemental grant once we found out some of the results from the initial study, and then discuss what we're working on now in terms of publications, and then talk about potential future collaborations that we're looking for. So let's start here. Okay, there we go. So the study goals, we wanted to use a non-human primate model to provide data related to chronic exposure to low-level domoic acid. We focused a lot on the toxicokinetics, thinking that one of the major issues with exposure studies in humans is to actually get a really good assessment of exposure. So with a non-human primate model, with the animal model, you can actually get a lot better, a fuller idea of what the dose response is for exposures. We also wanted to look at whether or not metabolism, excretion of domoic acid changes, if it was different based on whether or not you're exposed for the first time as opposed to chronic exposures. And then we also wanted to see if there were differences in kinetics with pregnant versus non-pregnant females. And then lastly, we wanted to look at what were the differences in kinetics of DA when you look at the maternal versus the newborn blood levels. But the major focus of the study was on looking at low-level toxic responses. We really focused a lot of our study on reproductive effects in the adult females. We did look at sort of general overt toxicity signs in the mothers, but we assumed that given our doses, which were 1/10 to even less than 1/10 of the toxic dose, that we wouldn't be seeing overt toxicity. And then again, the real focus of the study was on prenatal exposure and infant development. Okay, so let's go forward here. So in terms of the non-human primate model, we have a primate center here at the University of Washington, and I've done non-human primate work for all of my career. And we use the non-human primates on just selective types of problems that we feel are major problems for human health in terms of exposures to environmental contaminants. We do know that the toxic dose for DA is similar when you compare monkeys and humans, and there's a lot of characteristics of the primate in terms of their structure and function that mimics the human better than other animal models. So we chose Macaca fascicularis, which is a monkey model that's been used previously in several studies looking at DA exposure and effects. All right. So in terms of the original study design, we had 32 adult females that we separated into 3 different groups. We had 10 in a control group, 11 in a 0.075 milligram per kilogram, our lower dose, and then another 11 in our 0.15 milligram per kilogram per day. So those 2 doses sort of bracket the estimated acute tolerable daily intake for DA. As I mentioned, our exposure is our oral. A lot of the previous investigations in animals have been using IV or IP, so we felt that oral was the best route to go. They were dosed every day. We had clinical assessments of the adult females. They were bred with non-treated males, and we looked at various reproductive outcomes, and then looked at the neural development of the exposed and control infants. So this gives you a little more detail about the study design. I don't want to go through this. It's a fairly busy slide, but I think what you can tell from this is that it's when you're doing non-human primate work, it takes a long time to get these studies done. We had a 2 to 4 month period where we just trained the animals to do all the different assessments. We actually looked at all of our outcomes for 2 months prior to doing any dosing. And then we did a kinetic study the first day that we had dosing, and then another 8 weeks afterwards. We bred the animals, and it took from 1 to 7 months to get the animals pregnant. And then we dosed them, exposed them all through pregnancy, and did additional 2 kinetic studies during pregnancy. The additional studies that we did actually were on the postpartum period on the adult females, where we did some MR scans, and Rebecca will be talking about that. And then we have the outcomes from the infant testing. So Nina is going to talk about the different kinetic study results in her talk later on in the webinar. Okay, so results so far that we've published. If you look at maternal plasma concentrations, and as I mentioned, Nina will talk about this in more detail in terms of the kinetic modeling. If you look at this slide, if you look at the first column, we should have, it should be the first 2 on that column are the pre-pregnancy period, and then the next one is during pregnancy. And then you'll see that again for the 24, it's pre-pregnancy and pregnancy. So you can see that you see a dose response relationship in terms of the maternal blood levels. You see a lot more scatter in the higher dose than you do in the lower dose. And there weren't any really major changes across the time. So basically what you're looking at is the dots are each blood level from a female at that time of exposure. So this is sort of a scatter plot of all the data. If you want to see the summary, it's in the next table, where you can see that at the 0.075, animals were about 1 nanogram per mil in terms of the 5-hour sample, and about a little over 3 nanograms per mil for the 0.15. So a doubling of the dose more than doubles the blood level in the animals. And again, Nina will be talking more about the kinetics. All right, next. Maternal weight. This is at the weights of the animals during all the different time periods, baseline, pre-pregnancy, and then during pregnancy. And we didn't see any differences across the 3 dose groups in maternal weight. So there wasn't any effect of the DA at these doses on maternal weight or maternal weight gain during pregnancy. Okay. As I mentioned, we did some observations of the adult females throughout the study, looking for any kind of overt signs of toxicity, like seizures or vomiting. We did some work looking at visual swallowing and reaching and pickup. to look at motor coordination, and there weren't any differences in terms of the pickup and the visual orientation. We really didn't see any shaking or seizures or vomiting at these levels. So we didn't see any, very easy overt signs of toxicity. And as I mentioned, we weren't really expecting to see that. But what we did see when we were doing this reaching task, which requires the animals to reach out and extend their arm to pick up a small apple bit, we did see some slight tremors in the adult females. And I think the next one will show you these tremors. Okay, I hope we're all back. So that may have been difficult to see. If you look carefully, you could see that at the end of the extension, when they were just picking up the apple bit, you could see that there was a sort of a very high frequency fast tremor in the animal's hands. Okay. So what we looked at is that response over time, and these graphs that you're seeing show you that as the dose gets higher, the one on the right is the highest dose. We saw a lot more of the animals showing these signs. We had actually one control animal that was tremoring pretty much throughout the study, and we actually found later on, upon necropsy, that animal had a lesion, brain lesion. So if you look at the tremors, we looked at to see what kind of differences there were. And if you look at tests comparing the three groups, there's a significant increase in tremors for the highest dose group, the 0.15 group. Although we do see it in some of the animals at 0.075. So this was a surprise, and then we're going to have a little discussion about what we did in terms of some supplemental research looking at the basis for these tremors. Okay, in terms of maternal reproduction, we looked at what the number of breedings to conception, the conception rate, C-section rate, and the live birth delivery rate, and none of those were changed by DA exposure. Each of them had about 2 breedings to conception, but the variability across the animals in all the groups is quite high. The conception rate was quite high for all the groups, 90 to 100%. The live birth delivery rate was high for all the groups as well. We had one animal that was born in a breech position that didn't survive out of all the animals in the study. We did have a higher C-section rate for the animals. This was based on decisions that were made by our veterinarians at the primate center in terms of any kinds of signs that they feel that are indicating that there's some problem with delivery. If you look at the birth characteristics of the infants, we did see the normal differences in males and females, with the males being a little larger than the females, but we didn't see at these levels of exposure any effects on newborn size in terms of birth weight, crown rump length, and head size. We looked at their newborn behavior, looking at muscle tone, physical activity, skin color, using an Apgar test. One of the benefits of doing this research with monkeys is actually you can adapt different studies, different types of procedures that are done on human infants, and you can do them with the infant monkeys. So this is a common test that's done on newborn infants, and they're rated in terms of 1 to 2 on each of those things. So a perfect score would be 12. We did see some differences between the C-section and the vaginal delivered animals on this study, which is actually seen in human infants as well, due to the anesthesia that's provided during C-section deliveries. But that doesn't last very long. By the second test, everybody was pretty much the same. But we didn't see any significant differences between the domoic acid groups and the controls. So with these basic reflexes and their status at birth, everyone looked pretty good. During the neonatal period, during the first 3 or 4 weeks, there's also a test that they do with human newborns called the Brazelton neonatal assessment procedure. And we've adapted that for doing that kind of work, looking at reflexes and behavioral states, muscle tone, how they respond to different stimuli for our monkeys. And we did that every other day for 21 days. And we didn't see any differences between the domoic acid exposed animals and the controls in any one of these four activities that we look at. So they look pretty good during the neonatal period. So one of the first cognitive assessments that we look at, looking at visual recognition memory. And again, this is a procedure that's used with human infants. And it's been used in quite a few studies looking at different types of exposures in human studies, and has been actually sensitive to various exposures that people have looked at in the past. We've also used it in some of our previous studies, and it's a fairly sensitive indicator of how the memory is functioning in these early months. So how this works is that you show a stimuli to the infant, one on the left, one on the right, and they're the same. And then you can take that away and then show them the same picture again, and then a novel picture. And if everything is working well, the newborns, the infants will look at the novel stimuli. We actually show it again, changing the orientation or changing the direction of where the novel stimuli are, just to make sure there's no slide bias. So you can do this with either objects, or you can do it, if you look on the right side, you can do it with pictures of, for our tests, it's monkey faces. For humans, they use human faces. The human and monkey faces are much more difficult to scan and to remember than the objects. So it's a more difficult task. If we look at the results from this study for infant visual recognition memory, if you look on the left side, that's the visual recognition memory scores with using objects. And they're right about where they should be. They're 60% or over, which means that they all prefer the novel object, which means they basically remembered the object from the first time they saw it and could distinguish the novel one from the familiar one. If you look at the right side of the graph or the table, this is using the more difficult stimuli, the social stimuli. And the highest exposed infants were the only group that did not show a novelty preference using the more difficult social stimuli, which indicated that they basically couldn't distinguish the novel one from the familiar one. There wasn't a novelty preference. This could be due to a lot of different things. They may not have remembered it, or they may not have actually processed all the details of the stimuli during the first period that they saw it to know whether it was novel or familiar. So we saw some changes in early memory processing in the highest exposed infants. We're getting some feedback that the first video didn't work that well, but we're going to go ahead and show a second one. Okay, so I think we're back. We got some feedback that the first video may not have showed the tremors very well, but we did see some tremors, similar tremors than we saw in the adults, in a few of the infants that were from the highest exposure group as well. If you remember that video, the infants at this time were doing a reaching task, and they are uncoordinated. So you'll see a lot of uncoordinated activity, but those high frequency tremors that you see are not something you usually see. Okay. That's okay. I think we're running out of time. So we did see some of those tremors, and we only saw them for the first month or so. So basically, they kind of grew out of that as they got older. We didn't continue to see these tremors once they got over a couple months of age, but we did see them early on when doing our reaching task for the infants. Okay, So let's see where we are. Are we back? Okay. So I mentioned, given that we saw these tremors in the adults, we talked with our funding agency, which was the National Institute of Environmental Health Sciences, and we thank them for their support. We did talk with them about these tremors. We initially had not included any kind of post-evaluation of those animals, but we did get some supplemental funds, and Rebecca Petroff, my doctoral student, took the lead on writing the grant for that, and we did get the funds for it. So now she is working on this area of research for her dissertation. So she's going to take over and talk about her findings that she's had so far.
Thank you.
So, Rebecca.
So as Tom mentioned, we wrote a supplemental to mostly look at kind of the in vivo and also post-mortem effects of domoic acid in the adults. So that's where all my research is focused on. So the first study that we have been able to publish is on magnetic resonance imaging, MR imaging, and we used a type of MR imaging called diffusion tensor imaging, or DTI. And just really briefly, what DTI actually looks at is how water is moving in tissue. So it measures the directionality and the speed of how that water is able to kind of flow in tissue. And the concept behind this is that in the brain, we have bundles of axons in these really white matter major tracts. And what happens is that we can pulse a magnet in different directions to look at how this water is moving both along the gradient and against the gradient. And when we pulse it against the gradient, we'll have a pretty slow diffusion rate of that water, whereas we pulse it along the gradient, the water will want to flow with the axons. So we can use this to then measure how the tissue is structured in the brain. So this is all just a structural measure of in vivo brain white matter. So for this part of the study, we chose a subset of the adult female. We had a total N of 12 with 6 control animals, 2 in our low dose group and 4 in our high dose group. And all of the animals underwent a single sedated magnetic resonance scan with diffusion tensor imaging and a T2 weighted scan. And the T2 weighted scan just shows us a really brief image that we can look to see if there are any severe brain lesions. In the sea lion literature, we know that repeat exposures, especially at those higher doses that sea lions get, can lead to really severe brain lesions. So with that, we looked at this DTI measure, which again measures the flow of water in the tissue, as well as if there were any brain lesions in our animals who were tremoring. For the DTI, we used a cluster-based permutation and correlated this permutation with both dose and tremors. And one of the measures that we can use from this DTI is called fractional anisotropy, which measures the directionality of water, which was found to be significantly correlated with our tremor score. And just reiterating that what we found was a decrease in this fractional anisotropy, which indicates that there is less restriction of water, which suggests that the major white matter tracts are somehow decreased. And this could be for a number of biological reasons, some of which could mean an infiltration of brain immune cells like astrocytes or other glial cells. But it can also mean that the white matter tracts and the axons of those neurons are damaged too. So on the bottom here, you can see the image from our animals as to where we found significant differences in the brain. And I want to point out this major white matter tract kind of in the first 2 panels. All those little red and yellow dots are significantly different voxels, we call them, which are just individual little panels, each with their own unique numerical number that we can use to estimate this measure of fractional anisotropy. So this first major white matter tract that we see affected is called the internal capsule, which is a white matter tract that connects both the thalamus and the pons, and it holds a lot of your fibers that are really important for motor coordination, which makes sense when we start to think about our tremors and how we saw those. And then one of the other major white matter tracts that you kind of see in the fifth and sixth image down towards the bottom of the brain is called the fornix. And that particular white matter tract stems from the hippocampus, which is the major area of the brain that's affected by domoic acid when you have those really high doses of domoic acid. We also saw some smaller effects. So you can see kind of in the last two panels in the corpus callosum and the pons. And those are really important structures for motor coordination, but also connecting the brain across the two hemispheres. So this is a lot of data to look at. So if we just kind of look at the really big structures that we saw differences in, we can pull out those individual voxels and look at how the tremor scores actually relate to this DTI measure of fractional anisotropy. So for each of these graphs, you can see the tremor scores on the bottom, and then the fractional anisotropy is going to be over on your left axis. And you can see how the tremor scores are related to fractional anisotropy, both in the internal capsule, which is shown on the top two panels, and the fornix, which is shown on the bottom two panels. So that's just a really brief overview of what I was able to do with the MR imaging so far. And I'm going to then just talk a little bit about our working papers and what we're continuing to look at right now. So the first part, which is what I'll be really spending my dissertation focusing on, are continuing the research with the moms who are tremoring. So we have additional data that we're looking at with our MRI imaging, where we can actually measure the tracts and look at other measures outside of that DTI imaging. We can look at this tractography, it's called, where you actually look at how the tracts are connecting between different structures in the brain. And this is really important for understanding potential, not just areas that are affected structurally, but also kind of how the fibers are in a functional way. I'll also be looking at some EEG data. So we did in vivo EEGs on the moms as well, where we can look at some measures to look at how not just the brain structure is changing in relation to domoic acid and the domoic acid tremors, but also the brain function. So, and then the final thing that we'll be looking at, and we have these data kind of churning right now, is differences in gene expression given the domoic acid status of these animals. We're also working on some early infant manuscripts, including some data on early development up through about 1.5 years. So this includes measures on growth as well as socio-emotional measures and more data on cognition. We have EEGs from neonatal ages, including as early as 2 days, where we can actually go back and look at how the infants were sleeping and look at measures of REM and different quality markers there. And then we have EEGs on 1- and 2-year-olds, where we can look similarly to the adults about functional power in the animals that were exposed and not exposed. So I'm going to pass it back to Tom to just talk about some of our continued plans here, and then we'll wrap it up quickly.
Okay, thanks, Rebecca. That was great. So we wanted to just end with letting folks out there know that we do have actually some tissue from these animals that we would be very interested in collaborating with other researchers. We unfortunately weren't successful in renewing this grant We had proposed to do some additional functional assessments of the offspring now that they're getting a bit older, but that was not funded. So we're going to actually have frozen and fixed tissue from both. We do have right now the frozen and fixed tissue from the mothers. One of the things that we'd be interested is looking at the DA distribution across the tissue from these animals that were chronically exposed, or other outcomes and measures of DA toxicity on the adults. And then we will be beginning fairly soon necropsies of the offspring, and we'll also have frozen and fixed tissue for those to look at different parameters of toxicity in the offspring. So we just want to let folks know that we will be having a repository of tissue that we will be sharing with people that are interested. And I think that's it for us. Yeah, so acknowledgements. This took a long time, takes a lot of people and a lot of effort. There's a lot of students that were involved in this over the several years that this grant has been going on. And I'd also, again, like to thank the funding agencies that we had, both at the NIEHS and some local funding that we got. So with that, we'll take any quick questions, if there are any.
Thank you very much, Dr. Burbacher and Ms. Petroff. We do have a couple questions. The first question is, were the cage site observers blinded to the treatment groups? And then the second part of that question is there a way that the scorers were blind to previous scoring results? Perhaps you had multiple scores?
Yes to both of those. The scorers were blind, which actually increases the difficulty of doing studies like this, because you have to have separate staff doing the working with the adults and the infants, because all the infant testing was done blind as well. So yes, we did have blind testers who were blind to the treatment. We did have multiple testers, and but I mean there really isn't any way, unless I wouldn't know any way that you could actually separate multiple testing over time. So we did have different testers. The results were the same no matter what the tester was, but they all actually saw the animals over time.
Great. Because we do have quite a variety of attendees today with varying scientific backgrounds and some from the lay community, can you just briefly explain what you mean by blind?
Oh, sorry. That means they don't know what exposure the animals had.
Thank you.
So the people who do the exposing of the animals are completely separate from the people who do the testing.
Very good. A couple more questions for you. Were any of the fetal fluids tested for domoic acid concentration?
Yes, we did have some C-sections, so we collected some amniotic fluid. I'll let Nina talk about that. And we have some cord blood as well. And we have some newborn blood. So I think that's going to be more in Nina's discussion.
Sure. Another question, did the neurological effects resolve when the DA challenge is discontinued?
We did have a couple of adult females that we took off of the DA prior to necropsy, and those animals continued to show the tremors.
So just to clarify for others in the audience, that means that the tremors persisted even when they were no longer being actively exposed to domoic acid?
That's correct.
Okay. One last question for you. Does captivity have an effect on brain development? And if it does, how do you control for this in a laboratory setting?
Well, all the animals are nursery reared, so we're not comparing nursery reared animals to wild animals in the wild. There are some studies that would indicate that a nursery reared animal is not the same as an animal in the wild. But that's not the comparison. The comparison is for all the across all the infants that are nursery reared in this study.
Thank you. Very good. Thank you so much. We do have a couple general questions that I'll go ahead and address. One question came in from the audience asking about the 2017 workshop. That was an in-person conference, all-day conference at University of California, Davis, and unfortunately that was not recorded, so there is not a file with those presentations to share. However, the agenda is on our OWIHA website. If you look at owiha.ca.gov/fish, and then click on Domoic acid page, there is an agenda there. So if you're interested in seeing who those presenters were, I encourage you to visit our website. Another question came through asking about whether or not domoic acid is destroyed by cooking. So for just a general announcement, as we analyze the domoic acid levels in seafood, mainly done by the laboratories at the California Department of Public Health, the domoic acid levels are measured in cooked crabs. So the crabs and other species are prepared and cooked, and then the levels are analyzed. So those are the final levels in a cooked crab. It is also the recommendation of the California health agencies that when these seafood items like crab and lobster are cooked in water, that the cooking water that was used to boil and cook the crabs is thrown out and not retained for making sauces or roux or stews, because as the seafood is cooked, domoic acid can leach out or come out of the critters and into the cooking fluid. So it's a way to possibly reduce the level of domoic acid exposure if you discard the water. And so that's the general recommendation there. One note, the bivalve shellfish are not cooked prior to domoic acid testing, but the decapods, the lobster and crabs are. Okay, very good. Well, as we move on, our next speaker is Dr. Nina Isaharinen, Professor and Associate Chair of Pharmaceutics and Research Affiliate at the Center for Human Development and Disability at the University of Washington in Seattle. Her research focuses on pharmacokinetics and dose exposure relationships, including drug disposition, pharmacokinetic modeling, and drug-drug interactions. Dr. Isaharinen is presenting on the toxicokinetics and physiologically based modeling of domoic acid exposures. Welcome.
Thank you, and thank you for inviting me to be part of the webinar. I want to start with just acknowledging the team. Like Tom said earlier on, these things take a really big team, and especially with the various blinding strategies that we had on this study, we had a lot of people involved. So I want to acknowledge actually three graduate students, 2 graduate students and 1 research scientist from my laboratory at the Department of Pharmaceutics, Sarah Schoen, Jing Jing, and Ariel Toplets, who really were instrumental for developing the LCMS assays, doing all the validation, figuring out our dosing solutions for this study, that we basically QC'd every solution for every monkey throughout the entire study. And then really doing the modeling and the analytical work. And then the group that Tom already mentioned, who really were part of the non-human primate portion of the study and the behavioral assessments. And then obviously NIH for the funding that has supported the work. So what I'm going to talk about first is to cover really the fundamentals of the toxicokinetics of domoic acid and what we found out in this study. Using single dose administration, both intravenously and orally. And then go from there on how we use that data to develop a physiologically based model for domoic acid, first in the non-human primate, and then in the extrapolated that model to really predict what happens in humans and what the plasma concentrations and even brain concentrations might be in humans after oral consumption of domoic acid. And then finally, as the third part of this talk, I want to talk about what we found out in terms of the toxicokinetics during pregnancy and maternal fetal transfer of domoic acid in the non-human primate model. So just want to start with from a pharmaceutical scientist or pharmacokineticist perspective on how we look at domoic acid and its physicochemical properties. So it is a very polar compound, and part of why it's very difficult to work with as an analytical chemist is because it has a lot of ionizable groups and really has very poor permeability. So when we look at it as a compound, you would not expect it really to be absorbed from the gastrointestinal tract as such, because it really isn't permeable enough to get across the intestinal cells. What we found out also in our studies is that it does not bind to any plasma proteins. So all of the domoic acid that we find either in human or animal plasma is free or unbound, and it does not partition into the red blood cells. So when we look at blood or plasma concentrations, essentially all of the domoic acid is in the plasma water. And based on that, it is sort of surprising that after eating and consuming domoic acid orally, it gets absorbed, or we can find it present in the plasma. So we started by looking at some of the past pharmacokinetic studies that had been done in non-human primates. And the data that existed from the 1990s was following an intravenous dose of domoic acid. And what this figure shows is the serum pharmacokinetic or toxicokinetics of domoic acid after a 0.05 mg per kg dose of domoic acid intravenously. And what was found in this study was that domoic acid had a very short half-life of 1 to 3 hours approximately. And in the non-human primate, unlike what had been seen previously in rodents, about 60 or up to 90% of the majority of these animals, about 60% of the domoic acid was excreted into urine. What I'm noting here is that in the rodents, you pretty much find 100% of the dose, which is intravenously administered, in the urine. But in the non-human primate, it actually seems that there are other elimination pathways already based on this data. So one of our initial questions was whether this toxicokinetics that was established in the 1990s with the fairly high dose of domoic acid that caused acute toxicity would translate to the kind of doses that we looked at in the non-human primate model. So what we did is we repeated the intravenous study in 3 sort of what we call pilot animals. What you see on the left-hand side of the slide is the intravenous dosing pharmacokinetics. You may note that this is a 10 times lower dose of domoic acid than what was done in the previous study. So in these animals, we did not observe any acute toxicity. This is actually an intravenous dosing study that was done in 2 separate occasions in the same animals. So this is essentially a mean of 3 animals done twice in each animal. And what you see is that the half-life after the intravenous dosing is very similar to what was observed previously, 1.2 hours. What we saw when we looked at the urinary excretion of domoic acid in these animals was very similar to the previous study. About 40% of the dose was excreted into urine. So that's the AE in the table. Amount excreted into urine was 42%. What was very surprising, when the intravenous data really reproduced the work done earlier with much higher doses, what was really surprising to us was what we then found out in the oral dosing study. And what you see on the right-hand side is the oral dosing kinetics after the 2 different dosage levels that were ultimately used on the larger study that Tom just presented. 0.15 mg per kg and 0.075 mg per kg orally, given in a sugar water essentially to the animals. And what you see here is that when we give domoic acid orally, the half-life that we observe, what we call an apparent half-life, is actually much, much longer than what we observed after the intravenous administration. So you see that we see approximately 10-hour half-life in these animals, and we call this flip-flop kinetics. And partially why I emphasized the low permeability of domoic acid earlier is that this is not completely unexpected for a compound that is as polar and as ionized as domoic acid. What happens is that the absorption from the GI tract is actually so slow that it's slower than the elimination would be, and hence this half-life becomes rate limited by the absorption rate. And that is kind of surprising because it really changes our expectation of how the actual plasma concentration time profile is going to look like after oral exposure in comparison to what we would have predicted from the intravenous data alone. Another piece of information that we saw from here is that of the oral dose, 4% ended up as excreted unchanged in the urine. And from the data, because these are the same animals dosed both intravenously and orally, we could calculate the oral bioavailability of domoic acid as well. And in these animals, it was 6 to 7%, which is shown there in the table. So from these pilot animals, we actually gained a fair bit of information of the dose concentration relationship of domoic acid, meaning that we have a good handle on the bioavailability of domoic acid. We gained the information about the half-life, and we see the that there is a dose exposure relationship, meaning that when we give a higher dose, we also see higher plasma concentrations of domoic acid. So just to summarize the key findings from the single-dose PK studies, one of the things that we did observe is that after the oral doses, the toxicokinetics were very variable. The plasma concentrations, both within an individual animal and between animals, had a very high variability, likely due to the very highly variable oral bioavailability. The other very important finding was the flip-flop kinetics, meaning that after oral absorption, the elimination is absorption rate limited. And what that means is that we cannot predict the time to peak concentrations or the time course of domoic acid concentrations from the intravenous data. And the presence of domoic acid in blood is actually much more prolonged than what we would expect from IV data. But also that the peak concentrations after oral doses are going to be lower than what we would expect based on the intravenous administration. One of the interesting aspects that we found out mechanistically from this study was that the renal clearance or the renal elimination of domoic acid was only 30% of what we would expect from creatinine clearance. So this is in terms of the renal clearance being, we can compare it to the glomerular filtration rate of the unbound domoic acid, and we see that only 30% of what is filtered ultimately ends up in the urine. And what that tells us is that there is likely active transport that happens in the kidney that is responsible for reabsorption of domoic acid. And if that translates to human, it could actually have a major impact on the overall elimination of domoic acid in humans. We also found out that only about 30 to 50% of the dose was ultimately excreted in urine after the intravenous administration. And that really suggests that in the non-human primate, there are other elimination pathways for domoic acid. We did look at actually feces in these monkeys, and we did find domoic acid in the feces after intravenous administration, suggesting that there is biliary secretion in the liver of domoic acid. We don't currently know if the monkey here is the best model for human domoic acid elimination, but it's something that probably should be looked at if biliary secretion is relevant in the human situation, because that would also implicate that various conditions in the liver would affect plasma concentrations and elimination of domoic acid in humans. So with that, I want to move on to the next part, which is really the physiologically based modeling of domoic acid disposition. What you see here in the gray boxes is the standard physiologically based pharmacokinetic model. The arrows in the model really refer to blood flows going from arterial blood to venous blood, and we can essentially adapt this to either the non-human primate or to the human based on the physiological sizes and blood flows of these organs. It can also be applied to various different physiological conditions by changing, for example, the placental fetal unit that you see in the bottom, changing the expression level of enzymes in the liver, changing the kidney function, glomerular filtration, and so forth. So it's a very powerful method to predict changes in toxicokinetics in various physiological conditions. In terms of the domoic acid model, what you see on the right-hand side of the slide is the sort of overall list of things that we used for the non-human primate model. So we populate the model with the basic physical-chemical parameters, which help us simulate the absorption from the gut. We can actually put in the permeability values and predict how the absorption is going to happen. We include the blood to plasma ratio and then the partition ratios, which is referred to as KP values, into the individual tissues based on the properties of the drug to essentially predict how well domoic acid gets into the brain or all the other various organs. In terms of the liver and the kidney, in the liver, we used in the model the biliary secretion that we characterized after the intravenous dosing. And that's really, we populate that with the biochemical parameters in the liver. And then similarly for the kidney, we used the renal clearance values that we had gotten from the intravenous studies to simulate in this model the kidney clearance. And then based on that, we can also scale this using standard interspecies scaling from once we know what's happening in the monkey to human and predict what's the disposition gonna be look like in the human. So just to show you how the model turned out, for this modeling, we used a commercial software called Simsim that has a ready-built non-human primate model already there, so the physiology was set, and we really built the drug model in that platform, but one could adapt this model to any other PBPK platform that one would want to. And what's shown in these figures here in the bottom is the individual data points are on the left from our in vitro intravenous data, where we dose domoic acid intravenously in each one of the animals. And on the right-hand panel is the study that was done by Trulov and Iverson at 1994. So we did 2 independent intravenous domoic acid dosing studies with 10-fold different doses, and then simulated those. So the continuous line is our simulated mean, and the dotted lines are the 90% confidence interval for the plasma concentrations of domoic acid. And what you can observe from these graphs is that the physiologically based model really captured the intravenous dosing of domoic acid quite well. It is possible that in the 1994 study, the half-life of domoic acid is somewhat longer, but it's very difficult to compare these because one has to remember that our study was done with LCMS system and a different sample preparation than what was done in the original study. So some of this could just be assay variability from study to study as well. But what you can say is that overall, we are capturing the systemic toxicokinetics of domoic acid fairly well with this model. We then proceeded to test the model with our oral data. And this one shows you the 2 studies that we did after oral dosing of domoic acid. So again, each one of the individual symbols in the bottom panels shows individual data points on the 3 animals that we did the 2 different dose levels. So left-hand side is the 0.15 micrograms per kilogram oral dosing, and the right-hand side is the the left-hand side is the 0.075, and the right-hand side is the 0.15 dose level of domoic acid. And what you see here is that surprisingly also the mechanistic physiologically based model predicts the flip-flop kinetics and the longer half-life after oral dosing, just the same as what we see in the observed data. So as we look at the same 95% confidence interval and the mean simulated data, we can conclude that the model fairly well captures the oral absorption and the toxicokinetics of domoic acid. And what we typically would call from here is that our physiologically based model is now verified based on this. In addition, we took one of the data points that was one of the few data sets in the literature that have shown domoic acid concentration following multiple doses orally to animals. So what you see here, this is again from TrueLab from 1997. We captured the plasma concentrations from the published data. The individual circles on this show the observations of domoic acid concentrations in plasma, and then the continuous line that you see going through up to 16 days shows the simulated multiple dose kinetics of domoic acid in the monkeys that were studied in the 1997. And what you really see here is that regardless of whether we look at 20 years ago or today's data, the physiologically based model fairly well captures the oral plasma concentration time curves of domoic acid after the oral dosing, regardless of single dose or multiple dose. So we were fairly happy with the model at this point. The next question was then, because this is a physiologically based model, can we predict the brain concentrations or the acute toxicity using this model? And what we did is we looked at essentially the reported onset of symptoms after intravenous and oral doses of domoic acid in monkeys, and then tried to look at that duration of effect and the onset of symptoms, whether that matched to what a physiologically based model would predict in the brain. So what you see here is that after intravenous dosing, the toxicity symptoms start fairly rapidly within a couple of minutes from the dose. The duration of the symptoms is longer, the longer the intravenous dose, but still even with the 0.5 mg per kg dose, the duration is only about 2 hours after intravenous dosing that the animals will vomit and have the various CNS toxicities. In comparison, when you look at monkeys after oral dose of domoic acid, the onset of the symptoms is much later, as we would predict from the slow absorption, so from 60 minutes to 720 minutes. And then the duration can actually be longer, a fair bit longer, up to 400 minutes following the oral dosing, which would also be consistent with what we would expect from the flip-flop kinetics and the much longer half-life after oral exposure to domoic acid. So what we went and did is we simulated the brain concentrations using the physiologically based model. And what you see in this graph, the red line on the graph is the simulated brain concentrations in the following intravenous dosing that was used in the previous studies. The gold and purple lines that you see in the bottom are the predicted brain concentrations in our pharmacokinetic study after oral dosing, meaning that these were subacute doses. We predicted that the animals should not have any acute toxicities, and indeed when we look at the predicted brain concentrations, we would not expect to see any toxicities. And then the black line that is simulated is essentially the simulated brain concentrations of domoic acid following the oral dose that was observed to cause toxicities. And what you see, the two dashed lines on the figure, really are what we're looking at threshold of acute toxicity concentrations. What would be the brain concentrations that would need to be reached to observe acute toxicity? And then how long would the concentrations that we're simulating in the brain stay above that line? And indeed, what you see when you look at the time about the dotted lines, for the oral dosing, we would truly expect a much longer duration above the toxic threshold in the brain than after the intravenous dosing. So this was quite encouraging. And based on this data, we then went on and actually simulated what should happen in humans. Now, I do want to say that we do not have any data to really verify that any of this modeling truly applies to humans. We do not really know for sure certain that humans demonstrate the same flip-flop kinetics as we see after in the monkeys. But if we assume that the monkey predicts what happens in the humans, this is what we would simulate based on the physiological principles for human plasma concentration time curves after the 0.075 mg/kg dose. So the top panels are the plasma domoic acid concentration, and the bottom panel is the predicted brain concentration in the humans. And in the left-hand side, the panels A and C show the 0.075 mg/kg dose. And then on the right-hand side, what you see is actually doses that were observed in the first domoic acid toxication case that was discussed in the very beginning of the day. So these are what we are predicting should be the plasma and brain domoic acid concentrations in the individual that suffered from domoic acid toxication in the Prince Edward Island case that happened. I'm really showing this just to illustrate the feasibility of using physiologically based modeling and to give an idea of what we expect the concentrations in the brain or plasma would be when you take a acute toxicity doses of domoic acid versus what is currently considered relatively safe for domoic acid. So we can basically predict what we would expect to happen in humans. It really remains to be shown by some experimental data that this applies. So now to come back to then the real pregnancy study and the maternal fetal transfer of domoic acid, and I hope I will answer some of the questions that were asked earlier on about domoic acid in pregnancy and maternal fetal transfer. So I just want to show this one figure from the literature that shows essentially when domoic acid is given to pregnant rats intravenously, what is observed in the rat fetuses. And what you really see here, this is just to show that in the rat, domoic acid gets across the placenta, it gets into the fetus, and it gets into the fetal brain. And we also see a fairly long-term exposure in the fetal or long-term concentrations in the fetal brain after the IV dose. Now the question is, can these findings be extrapolated to lower doses or longitudinal exposure? So this is just to summarize again the overall study design for our pregnancy study that Tom already showed. Really, the overall study design is running there on the golden line. The red ones are the ones that I'd like you to focus on for the part of my talk, which means that we basically did a first day in before these animals got pregnant, a toxicokinetic sampling, which was a full 24-hour pharmacokinetic study after oral dosing, and collected urine as well on that day. Then 56 days later, we did another full toxicokinetic study for 24 hours, which is our pre-pregnancy baseline steady state domoic acid toxicokinetics. The animals were bred after that, and then as they got pregnant, we did on the second trimester, third trimester, and postpartum, 6 weeks postpartum, another full 24-hour toxicokinetic study. And in addition, at delivery, thanks to Tom's wonderful team, we got maternal and infant sampling at delivery, so I'm going to show you some of those concentrations. And then we also analyzed amniotic fluid and so forth that was mentioned already before. So here is the overall toxicokinetics observed in the pregnant and pre-pregnancy animals. What you see here clearly is that the plasma concentrations are higher with the 0.15 mg/kg dose than the 0.075. You see that as we go along from the pre-pregnancy to second trimester and third trimester, there is some level of decrease in the AUC of domoic acid. And what you see when these animals are chronically exposed, that essentially we see exactly what we would have expected from the flip-flop kinetics, that there is almost no fluctuation on the concentrations of domoic acid over the 24-hour dosing interval. What you see is that essentially the concentrations increase slowly after the dosing and then stay high across the dosing interval. And this is a very busy table, but essentially summarizes all of this pharmacokinetic data that we collected. Shows you that by the third trimester, the AUC actually is significantly decreased after the 0.075 mg/kg/day dosing group in comparison to the pre-pregnancy. So the top part of this table is the low dose, and then the bottom part of the table is the higher dose of domoic acid. So we see that the oral clearance is increased at the third trimester compared to postpartum. And this is most likely due to the increase in the renal clearance of domoic acid. We would expect that, based on what we know both in humans and monkeys, that the kidney functional filtration goes up significantly during pregnancy, which we see in the creatinine clearance values in this table. So we see at least a doubling of the creatinine clearance, and that proportionately translates to increase in the renal clearance of domoic acid. We also see that the fraction of domoic acid excreted and changed in the urine is very comparable between the two with the pilot data that we did. One of the things that I do want to note is that there is some differences between the lower and higher dose in this table. So if you look at the very rightmost column, which is the fraction excreted and changed in terms of the percentage, you see that it looks like it's about twofold higher, the fraction of domoic acid excreted and changed in the urine in the high dose group compared to the low dose group. Similarly, when we look at the oral clearance or the dose normalized AUC, we see that that is actually significantly decreased in the high dose group compared to the low dose group. So that is the third column from the left, that is the clearance over F. If you look at the third trimester number for the low dose group, it's 420, and in the high dose group, it's 190. So we see about 50% decrease in the oral clearance going to a higher dose. It seems that that is probably, our hypothesis is that that is probably because of some impact on the absorption with the higher doses, but we do not have enough mechanistic data to explain what really is the driving force for that. However, by and large, the variability in the domoic acid kinetics within and between individuals is larger than what the observed pregnancy effect in the changes in the kinetics is, suggesting that really non-pregnant individuals can be used to predict what happens in terms of pregnancy to the toxicokinetics of domoic acid. Now, last part then is what happens to the maternal fetal transfer of domoic acid and the fetal concentrations. And what you see here on the left-hand side is the concentrations in the infant plasma at delivery. And on the right-hand side is the maternal plasma concentration at delivery. The numbers given, so each one of the dots is an individual animal in both of the groups, the low dose and the high dose group. And the numbers above give you the average concentration in the group. So if you compare the 0.075 dosing group on the left panel to the right panel, what you see that the infant plasma concentrations are a fair bit lower than the maternal concentrations, and the same applies to the higher dose group. In addition, what you see is that the higher dose group, also the infants had higher plasma concentrations at the delivery. Now, what is somewhat surprising is that after the higher dose, the 0.50 mg/kg/day dosing, we saw a significantly higher maternal to fetal DA concentration ratio than what we saw after the lower dose. So the maternal to fetal DA concentration ratio is 0.6 after the high dose and 0.3 after the low dose. And it is possible that there is some transporter function in the placenta that contributes to that ratio, but this study was not designed to test dose-dependent differences in the maternal to fetal concentration ratio, so I would not read too much into that. And based on these data, we can also predict, putting together a physiologically based modeling of this data, that the fetal brain concentration should actually be 40 to 70% lower than what we observe into maternal brain, because the placenta does actually limit the exposure or the passage of domoic acid into the fetus. So consistently, the fetal or infant plasma concentrations are lower than what the maternal plasma concentrations are. So with that, I'll conclude. So just to summarize all what I've said, so number one, I think one of the big important findings that we had was the flip-flop kinetics after oral dosing. That really does affect the shape of the plasma concentration time curve after oral exposure. Another important finding was that really what was found about the domoic acid elimination pathways in the rodents didn't entirely apply to the monkeys. What we found is that only part of domoic acid is cleared by the renal route, and we really expect that biliary secretion is an additional clearance mechanism. We tried some metabolic studies as well, but could not find any metabolism of domoic acid, either conjugation or oxidative metabolism. One of the really key findings that I hope I've alluded to is that all our data from this study, whether it's the intravenous dosing, oral dosing, the pregnancy, or the maternal-fetal or maternal-infant ratios, suggest that active transport really plays a very important role in domoic acid disposition. We have some hypotheses of what the transporters at play may be, but I think we really need to invest some time and effort into identifying the critical transporters for domoic acid so that we can understand which species really will translate to humans and what the relevant elimination pathways are in humans. One of the things that I hope I explained is PBPK modeling can really be used to predict the domoic acid concentrations in humans following different exposure scenarios, both low dose, high dose, single dose, multiple dose, and so forth. But I think we do need some data from human plasma concentrations after known exposures to verify the model. In terms of the DA absorption, it's very variable, and I think that will really contribute to variability in individual responses to domoic acid, and the pregnancy effect is less than that variability between individuals. And then the saturation of DA transport is a possibility that it actually plays a significant role in dose-dependent kinetics of domoic acid, but we don't have enough mechanistic data to elaborate on that. And with that, I will conclude, and I'll be happy to take any questions.
Thank you, Dr. Shannon. We have a couple questions for you. So you've talked quite a bit about the variability that you've observed. Can you elaborate just a little bit more on that and its relationship to the absorption and kidney clearance and reabsorption? And then what might that imply for sensitive humans?
So in terms of the kidney clearance, what's quite surprising is when we do the intravenous dosing, there's very little variability from animal to animal. And if you look at even the data from 1997 or 1994 and what we did now, the intravenous kinetics is incredibly reproducible. In comparison, the oral absorption is really, really variable. So I think majority of the variability we observe comes from the oral absorption. We don't know if that has to do with ionization status, whether that has to do with, we actually did some pilot studies with fed versus fasted, for example. We never really observed a food effect, for example, in the monkeys. Doesn't mean that that wouldn't happen in humans. So I think that part of variability is going to be enormous. How the domoic acid, both the rate and the extent of absorption from the GI tract. That doesn't mean that if an individual has renal impairment or kidney disease, or in my opinion, we should probably even think about liver disease based on the findings on the biliary secretion. That's going to have a big impact on inter-individual variability in the clearance. We did not have that in this study because we did not have monkeys that would have had an organ impairment per se. So I think in a human population, that's going to contribute significantly to inter-individual variability. So if somebody has a declining kidney function, that is going to contribute to much greater plasma concentrations per dose than in a healthy 25-year-old individual. Does that answer your question?
Yes. So in tandem with that, we noted that the IV dose levels were significantly lower than those used for the oral dosing. The question is, how do you reconcile the two?
So we basically did not want to have any acute toxicity in the animals. And if you imagine that in the oral dose, only 3 to 5% actually ends up being absorbed, we basically wanted to have roughly the same plasma concentrations after the oral dose as after the intravenous dose. Because if there is any kind of saturation ongoing, for example, you want to have the same concentrations to compare one to the other. So we had to a priori adjust for that expected 95% of the oral dose that does not get absorbed. And that's why we used a much higher dose orally than intravenously to essentially achieve the same kind of plasma concentrations.
Thank you. Next question. Although domoic acid has multiple charges at physiological pH, is it passing through the blood-brain barrier? Does that mean that amino acid transport is used to cross the blood-brain barrier?
That's a really good question. You know, when you look at the molecule, you really wouldn't expect it to get to the brain. It was surprising to us that when we do the physiologically based modeling, we actually do not include any transport processes in that. So when we simulated the brain concentrations in our modeling, we did not include any active transporters. So some of it can actually get across regardless. I don't think that it's impossible that there is an amino acid transporter. I think in particular, I would probably expect that to happen in the intestine. So I think there is a very real possibility that there is, in addition to active transporter, and we would really need to do some brain concentration studies to get a good handle of what the true brain concentrations are. And I could even envision that at some different parts of the brain, there's going to be different concentrations of domoic acid due to different expression of transporters and so forth. So our data cannot refute or verify that there is a transporter.
Okay, very good. Another question is, are the offspring from the pregnancy study available for MRI, and will they be studied? Or are these monkeys shared with perhaps the Burbacher work?
These are the same monkeys that were done in the Burbacher work.
Very good. Okay, another question. Can you speak to the number of subjects, both maternal and fetal, that were lost to follow-up and how you accounted for that?
What do you mean that were lost to follow-up?
Not sure. Perhaps that person can send in a clarifying question.
Because these are the same monkeys that were followed then in all the studies that Tom just presented. So I don't think we lost any monkeys to follow up. We did the fetal, like the infants, the blood draws were done later on. So we have actually, you know, followed their domoic acid concentrations up to, I believe, a week after birth. We can detect the domoic acid in some animals, but we did not lose anybody to follow up.
Okay, she clarified, and she said that, for example, in the event of an unplanned death or a miscarry.
Oh,
or any, did that occur?
We did have one female deliver a breech baby, which died during the delivery.
Okay.
So that's the only one that was lost.
Thank you. Looks like a final question here. Is it possible that DA accumulates at sites within the body so that not all of the dose is eliminated?
Theoretically, it is possible that it accumulates somewhere, and that's why we recover only 40 to 50%. I think there is a long-term elimination phase for the DA. We actually did a study that I didn't present where we followed urine. We collected urine for, or I should say Tom's group collected urine for weeks after the last dose in an animal. And we could see the domoic acid for really, really long time in the urine samples. It's incredibly low levels, so I don't think it will ever account for the 100% of the dose, but I think there is a portion of the domoic acid that sticks around for a very long time.
Thank you very much. Not seeing any further questions, we are going to take a short break. We're running a little bit ahead of schedule. All right, everyone, welcome back. Our final speaker was also a featured presenter and panelist at our previous workshop. Dr. Lynn Gratton is a professor of neurology, epidemiology, and public health. She's the director of the Neuropsychological Diagnostic and Research Laboratory and director of clinical neuropsychology at the University of Maryland School of Medicine in Baltimore. Her research examines the neuropsychological impacts of exposure to marine harmful algal bloom-based toxins, including a key partnership with Northwest-based Native American tribes for the epidemiological study known as the Coastal Cohort, which she will now describe. Welcome, Dr. Gratton.
Lynn, I believe you're self-muted. Lynn, you may need to go to your control panel and unmute yourself.
Oh, sorry.
Thank you.
Okay. And this is Lynn Gratton, and it's an honor and a pleasure to have the opportunity to share our work with you today. And when I say our, I truly mean our. There are a number of collaborators involved in this study. And they're listed on this slide. The main findings of the study that I'll be discussing at the end have been published and in Toxins. And I'd also like to acknowledge the 2 NIH-funded projects that helped us bring these studies to conclusion. Okay, first of all, I'd like to just give you a brief overview of the background and of the study and what I'll be presenting today. First, I'll be discussing the general background. Secondly, I'll briefly summarize the overall cohort studies through which this initial study has taken place. And finally, I will get to the meat of the talk, which is the specific impacts of exposure on everyday memory. Okay, we all know from our earliest presentation today that high levels of domoic acid is neurotoxic. It's neurotoxic to humans, to wildlife, to sea lions, and in Dr. Burbacher's laboratory, it's neurotoxic to monkeys. The greatest exposure risk to humans in the Pacific Northwest is through razor clam consumption. And this is largely because in the razor clam, the toxin, domoic acid, accumulates through feeding and then is extremely slow to depurate. A crab may depurate the toxin out of its viscera within a couple of days. A razor clam can hold the toxin in its meat for up to a year, regardless of whether or not it's been frozen, canned, or otherwise preserved. and it might even hold it longer than a year, except Dr. Jackelkel, who did these studies, concluded it at the end of 1 year. So that makes the razor clam perhaps the riskiest potential source of domoic acid exposure in the Pacific Northwest. Currently, as previously discussed and described, the regulatory levels for harvesting shellfish is 20 parts per million. And in the 12 years during which I've been conducting all of these studies, there have been no reported cases of acute poisoning in the cohort I've been studying. Now, while we know there's been no acute impacts of exposure, of a single acute exposure or severe illness, the impacts of repetitive or chronic low-level exposures remain unknown. Therefore, the overarching research question for all of our studies is whether or not is repetitive or chronic exposure to low levels of domoic acid by a razor clam consumption associated with memory decline. I think I may need someone to take over the slides.
Certainly, Lynn.
Thank you. Next slide, please. So right now, I'm going to jump into the summary of our cohort studies. Next slide, please. Basically, razor clams are a very valuable natural resource to the Pacific Northwest tribes. It's an important part of Native American history and culture. It's a source of protein in their diets. It's important to the local economy as they distribute these clams, and they can them and harvest them. They sell them all over the world. It's also important to tourism, with between 250 and 400,000 harvesters a year come to the Washington coast to try their hand at digging razor clams. And once again, even the non-Native American industry is in many ways dependent on their ability to sell their razor clams. Next slide, please. Okay, razor clams of the Pacific Northwest are eaten quite regularly, and when you're there, you can have them for breakfast, as you can see on the far left. Lunch, you can have a cup of soup for lunch, and dinner. Now, these slides are taken from meals at a fine dining restaurant associated with one of the tribe casinos. Not everybody has their clams prepared exactly this way, but these they can be quite appetizing and delicious. Next slide, please. Okay, now let's talk about amnesic shellfish poisoning, which was the syndrome named and associated with the major outbreak in Montreal back in the late 80s. Now, the memory falls on a continuum in humans. You can become totally amnestic, which was many of their patients were, which means pretty much everything goes in 1 ear and out the other, sort of like a replay of the movie called Groundhog Day. They learn something new, and 20 minutes later, they completely forget it. That was the amnesic shellfish poisoning. Now, memory, people just don't go from normal memory to full amnesia. They can have mild impairment, moderate impairment, etc., as we can see oftentimes with people with dementing illnesses. Next slide, please. There are a number of factors that potentially impact memory, and let's just keep clicking ahead on slides, please. Age can affect memory. Education can affect memory. Occupation, someone's work, occupational status can affect memory. Their sex, gender can affect memory. Their marital status, believe it or not, also affects memory. Their medical history, other medical factors can affect memory. Fatigue, being tired, can affect memory. Drug use can affect memory. Depression can affect memory. Anxiety can affect memory. Generalized cognitive status can affect memory, and alcohol abuse can affect memory. Continue, please. And drug use, of course. Next slide, please. Oh, and effort. Definitely effort. When we're doing our exposure assessment, please go back one. When we do our exposure assessments, basically we do a dietary assessment. We look at what they've been eating, and we developed and validated a shellfish assessment survey. Basically, this is a measure of how much shellfish, how many razor clams people are eating on a daily, weekly, monthly, or annual basis. We validated this measure against a couple of standard measures, the block dietary questionnaire, as well as 24-hour food diaries, to make sure that we were capturing the information we wanted to capture. We also looked at number of clams consumed by average monthly DA levels at particular source beaches. This latter method was a little bit too vague in general for us, so we did not use it in our findings. Next slide, please. All right. If you see the black oval on this graph, and this is basically some data on the Washington State maximum on domoic acid from the years 1991 to 2015, provided by a Dr. Vera Trainer from Northwest Fisheries Science Center in NOAA in Washington. And in the black oval area, these were the DA levels at the time of our study. Interesting what sometimes happens when you have a study in the field, the toxin does not appear. But of course, we were asked to come in and look at this in the tribe after the higher levels, as you can see, the elevated levels on the right, on the left. Now, if you look at the right, this is when we begin to have what was called the big blob that was talked about earlier. After the blob, in the far right line and dot, the levels started to come back down. I just want you to keep in mind that as these levels were falling just below 20 parts per million was when we conducted our everyday memory surveys. Okay, next slide, please. All right, I'm just going to summarize the findings of the epidemiological studies we did with the tribes. Overall, it was a 12-year study with data analyzed at 4 and 8 years. We saw over 1,000 participants, or did 1,000 exams during this time, with about 500 to 600 actually sticking with us throughout the entire study. So it's their data that we've analyzed and reported. Now, these individuals were randomly drawn from tribe registries. They were cleared for a general intellectual status, and also we made sure that they could see the stimuli properly and they could comprehend it properly. We found and we saw them once a year for the time of the study. And we found that if people ate 15 or more razor clams per month over 12 consecutive months, after 4 years, we noted a mild memory decline. After 8 years, we continued to have the exact same finding. So we kind of replicated the study ourselves. Within that context, I'd just like you to know that 22% of the Native American cohort would be considered high consumers by this standard. Just over 20% would be considered, let's say, at greater risk of having mild memory decline after multiple years. Next slide, please. Now, this is the big question. What is mild memory decline? Well, it's slight. We can measure it. But let's keep in mind that in a neuropsychology laboratory, you can measure just about anything, just depending on the sensitivity and specificity of your measures to what you're looking at. So yes, we measured it, and we found that it was a slight decline. However, whether or not it actually impacts daily life or is noticeable to the person, to the individual, we're yet to determine, like, is it meaningful? Next slide, please. So right now I'm going to talk a little bit about the impacts of the domoic acid exposure on everyday memory. Next slide, please. The overall research question for this particular study is, do the mild memory changes found in the domoic acid cohort over time actually make a difference in the daily lives of the coastal dwelling Native American people in the Pacific Northwest? Next slide, please. So now, what is everyday memory? It's another construct that we actually have to begin to measure. Everyday memory is essentially functional memory, or the frequency of memory failures in everyday life. For example, how often do you have to check something that you should have done? How often do you forget to do things that you should have done? How often do you forget something that you were just told yesterday? How often do you forget to tell someone something that is important? Now, we all have some memory failures on a day-to-day basis. Most of the time it's due to inattention or stress or lack of sleep. But in this study, next slide, please, we took a look at this using a standardized everyday memory questionnaire. Now, in this particular study, it's a sub-study of the cohort. We looked at 60 adult men and women ages 18 to 79 after a community harvest. This was the first community harvest after the opening of the beaches of the blob, which means after they've been closed for months due to elevated DA levels. DA levels around that time were not 3 to 5 parts per million. They were more like 8 to 14 parts per million. Okay, we administered the everyday memory questionnaire. I gave you a few examples of that questionnaire in the previous slide. And the reference time period was since your last razor clam meal. So basically, they were asked to think back to their last razor clam meal and respond to the memory questions accordingly. For exposure, we simultaneously collected data on using the Shellfish Assessment Survey. We looked at exposure in 2 ways. We looked at exposure for the prior week, because it was up to 7 days that they could have had their last meal right after the harvest, or the past year. What was their exposure over the past year? Thinking perhaps we may find a difference between target week and past year memory problems. All participants were reimbursed $25 for coming in for our assessments. Next slide, please. In addition to having 2 time periods, target week and prior year of exposure, we also divided the group into exposure levels prior to our data analysis. Low exposure were considered those folks who consumed between none or 14 razor clams over the past 12 consecutive months. The high exposure groups were participants who consumed 15 or more razor clams over the past 12 consecutive months. The 15 cutoff was based upon the data from the prior cohort study that indicated that people who ate 15 or more clams may have a memory problem. The memory problem we found was mild. Next slide, please. Okay, and we had the exposure time frames, as I noted before, but I'll review again. Once again, with a number of clams consumed within the week or 7 days prior to the assessment, and the past year number of clams consumed over the prior year. And here you see a picture of clam fritters. Our preparation methods were taken into consideration with the amount of clams consumed adjusted by our dietitian on the project after a lot of cooking and making fritters and making chowder in terms of how much clam would actually be in each portion. Next slide, please. Okay, and this is our first data slide, and it's quite busy. And I'll have a summary follow-up slide immediately following. In any event, what you can see is we've got an average age of 42, slightly more women than men. And when you come right down to it, the high DA exposure group had more memory problems than the low exposure group, whether it was the target week or the prior year. And let's go on to the next slide where we can just look at this in summarized form. Okay, so we've got the average age of women in the sample was 42 years, or the average age of the sample was 42 years, pardon me. 56% were women, 51% reported at least one or more everyday memory problem. Let's keep in mind a higher score on the everyday memory scale means more of a problem. There was no significant difference in age, sex, or education between the DA groups or between exposure groups at either time epoch, whether it's the target week, the week prior, or the past year. So whether it was a high DA exposure group or the low DA exposure group, they were not in any way different on these basic demographic variables. We did find, just calculating descriptive t-tests, that the high DA exposure group had higher everyday memory problems and were more likely to report any memory problems than the low exposure group for both the target week and the past year. Of course, with that statistic, we did not take into account all of the possible intervening variables. Next slide, please. And this is where we begin to take into consideration age, gender, and education in this data. And as we can see on this slide, we're beginning to see a trend in education, but it goes in the inverse direction that you would think. People with higher education reported more memory problems, but we're going to be discussing that closer to the end of this presentation. Next slide, please, so we can summarize all of this. Okay. DA exposure from the prior slide indicated that during the target week, it was not associated with everyday memory score. That means how many clams they ate the prior week after controlling for age, gender, and education had nothing to do with the everyday memory score. If you do a linear regression and use your outcome and use everyday memory as a continuous variable for the outcome, the everyday memory score was associated with the participants' DA, domoic acid exposure, or razor clam exposure during the prior year. So the week before did not appear to affect their everyday memory score, but the amount they ate over the prior year affected this score. We also found that higher exposure during the past year had everyday memory scores that were higher than the lower exposure participants. Next slide, please. And here we're going to see another busy slide. Basically, as we can see, there's significance at both levels for target week and past year in our models. And this is a logistic regression model where the outcome is high everyday memory or low everyday memory based upon a median split of scores. Next slide, please. Okay. Now, in this logistic regression, we looked at the association between domoic acid and the odds of having any high or having a high score. And we found that high consumers of razor clams, that's more than 15 a month, were 5 times more likely to have more memory problems than the low consumers, whether you were talking about the prior week or the prior year. Okay, so high consumers are 5 times more likely to report memory problems or be in the high memory problem group than low consumers. Okay, next slide. And this is the last logistic regression table where, again, we're finding significance at target week, and that impact of education is rolling in there again. And this is where we'll get a chance to talk about that. Next slide, please. Okay, so in summary of the previous slide, looking at the presence or absence of any memory complaint, we found that high consumers over the target week were almost 4 times more likely to report any problem at all than the low consumers during the week prior. There was a trend for people with higher educational levels to be 3 times more likely to report at least one memory problem than high consumers. And this finding sounds maybe counterintuitive or paradoxical, but the explanation for that is that reporting the number of memory problems that a person has on a daily basis requires an element of what we call meta memory. That means being able to extricate yourself from where you're sitting right now cognitively and reflect back on what's been going on with your memory for the last week. And available data has consistently suggested that awareness or insight into the memory problems that you've had over the last week is associated with education. So then it would not be surprising to find that people with higher education have the tendency to report more memory problems, though it never reached significance when controlled for in the regression procedures. But it certainly is worth noting. Next slide, please. Okay, so in general, I'm just going to summarize all of the everyday findings at this point with this slide. And that is that high consumers, both the target week and the past year, were 5 to 6 times more likely to have an elevated everyday memory score. High target week consumers, that means high consumers right after the dig and before we tested them, were more likely to have at least one memory complaint compared to high razor clam consumers over the past year. Is one memory complaint meaningful? Well, statistically it was, but they did have one more complaint than the overall year. And then there was a trend for people with higher educational levels to be 3 times more likely to report at least one memory problem than those with less education. Next slide, please. As a result of this study, the findings for this study were shared with the State Health Department and the tribes prior to publication. The Washington Department of Health put out a statewide razor clam advisory, basically advising people in general to not be eating more than 15 clams a month, and specifically targeting vulnerable groups: pregnant women, children, and elderly, largely because we suspected there may be some special problems with this group during in utero exposures, and possibly with older people and renal clearance. Though I'm going to defer to Nina to discuss those possibilities. But these groups we did choose to take into consideration in that. Next slide, please. I'd like to review some of the limitations of this study, because in human studies and human epidemiological studies, there's far less control over exposure and measurement than in the lovely laboratory studies I presented before me. Basically, we don't know their historical exposures. We can ask them how many years they've been consuming razor clams, but those memories will not be as reliable as, let's say, the prior week, the prior month, or the prior year. So they may have had a combination of high and low level exposures throughout their lifetimes. Secondly, these findings may not generalize to infants toddlers, or geriatric populations. Thirdly, the findings may not even generalize to non-native populations. And finally, there's always an issue of recovery. Do they eat a lot of clams and then they get back to baseline, and then they eat a lot more clams and they get back to baseline? And once again, some of these questions are best answered in a laboratory settings. Studies by Kathy Lefebvre suggest that there is some recovery. However, we need to follow up as Dr. Azira had and find out what her studies are contributing and will continue to contribute to this knowledge. Next slide, please. Okay, future directions or places that we'd love to go. We'd like to do extended studies of vulnerable groups, studying infants, children, the elders, and seniors as separate and individual cohorts and be able to study them robustly. We believe we need to take into account some of the cultural aspects of exposure. So studies of non-Native Americans would be a great idea. And just to add, we just wrapped up a study of non-natives in terms of a generalized risk assessment, just to see how many clams they were eating. Do we really need to worry about the general population of Washington State raised clam harvesters being affected by eating a lot of clams? I mean, how many actually eat 15 or more a month? We know 22% of the Native Americans do, but do the non-native recreational harvesters eat that many? And our findings, which we just ran calculations on within the last 2 weeks, was that about 16% of licensed razor clam harvesters eat clams at what we consider a high level, 15 or greater a month. Now, we reached the most heavy consumers in the state because they're the actual razor clam harvesters, and we had caught them right off the beach. As they were coming off the beach, we did a random sample of them. So they would be expected to be the highest harvesters in the state. In comparison, Dr. Ferris et al. In toxins found that about 3% were high consumers who were exposed to clams on the Washington beaches. The difference in the studies is that in the study we conducted, we focused on the highest consumers, which is the harvesters, and took them right off the beach. In the other study, data was collected from people recruited off the beach to mail in surveys reflecting up to 6 family members. And so when you're adding in all the family members, in addition to the harvester, you may end up with fewer people eating a high number of clams, or possible more just vacationing harvesters. Okay, we also need to take a look at the reversibility, and I'm really not sure this can be done in human studies, at least not ethically. I think we need to look to our animal modelers, such as Dr. Burbacher, to answer, and Dr. Kathy Lefebvre's questions, because I think it would nearly be impossible to have a person eat clams and check their memory and then eat clams again and we check their memory again. Also, we don't want to be encouraging people to be eating over time a clam that may be toxic, or that we know is toxic to some people. And we're also working on the best way to promote effective health messages for prevention and management if these are indeed a slight memory problem. There certainly would contribute to a different message than if we were worried people were all going to go around and be amnestic if they ate too many clams. Finally, to be able to better assess exposure, the only way and the best way we could assess exposure to domoic acid was through number of clams consumed in their diet, and it's a self-report of that. Now, we have no reason to believe that this group, we ran analyses for outliers, was doing any exaggerating or minimizing, but we certainly don't know exactly how much they were exposed to. So identifying a biological marker that could be applied in humans would be the best, the ideal way to move forward with looking at whether or not there's a direct relationship between the memory problems and exposure. Now, the first step of this was completed by Dr. Lefebvre et al., including Nina Izirahan, who spoke earlier, spoke right before me. And there was a large group of chemists and biologists involved, because while I was collecting the everyday memory data from folks, we were also collecting blood and urine samples. Those samples went back with Dr. Lefebvre, who blinded colleagues and sent them and published the paper basically saying that they found a potential blood serum antibody associated with the DA exposure. This is in no way yet ready to become a marker, but I need to thank Kathy Lefebvre and her colleagues for at least starting that work, so we can look forward to that in the possible future. Now, I am finished at this point with my formal talk. I'm done a little early. I'd be happy to answer more questions or turn it over to the whole group for questions regarding this entire webinar. Thank you very much.
Thank you, Dr. Gratton. We do have a few questions from the audience. Several of them had to do with reversibility and looking at recovery, and some mentioned the zebrafish and mouse work from Dr. Lefebvre's lab. I think you've covered that. Another question comes here. Do these memory loss issues, are they permanent or temporary in the population that you've observed so far?
That I can't answer. That has to do with recovery. Like we found over 4 years and 8 years, we saw mild memory problems occurring over time. They never reversed back to not having a memory problem. But I cannot speak to that recovery, because as I mentioned, it would be too hard to measure in any detail with humans.
Thank you. Another question comes from, as with looking at any correlation with DA exposure, oral DA exposure, and increased incidence of other dementia-like symptoms and syndromes such as Alzheimer's.
Not based upon our cohort. We would like to look further into that and just study a cohort of elders. However, based on our cohort, there was no evidence of general cognitive decline, and we used comparison measures of constructional praxis, language, and attention and concentration in our analyses, and none of them showed any decline. They were all stable over the time of the study. That's an excellent question, and that's a direction we need to go in at some point in the future.
Sure. Question comes from, what toxins were the clams tested for, and were any of these participants tested for the toxins as well? You'd mentioned that there were some biological samples taken. Do you know what the DA levels were in these clams that were harvested, or can you speak to that perhaps? What is known about the concentrations of the clams that were consumed?
Our best available knowledge are the reported concentrations that are established by the Washington Department of Health and then reported on the website from the Department of Fish and Wildlife, and we stay in close touch with everyone after the studies are over. We're blinded during the studies, that meaning we're unaware of the DA levels during the studies, except the last round of study we did. We knew the levels had to be below 20 for us to go out and test participants because they were eating. So basically, we believe the levels were somewhere between 8 and maybe 14 parts per million.
Okay. Do you know if the razor clams were tested for any other contaminants, including mercury?
I do not. However, what I can tell you is that we took 20% of our large cohort sample and got hair mercury levels established, and sent them in to be tested for mercury, and it was considered they were found to be having not a significant exposure to mercury. And that was through measuring the hair samples of the participants.
Okay, thank you. Another question comes from the audience. Are razor clams the only significant source of domoic acid for human populations? Are there other shellfish that may not hold onto domoic acid as long as razor clams, but are eaten more readily and could pose a risk for exposure?
I think at this point, the greatest risk for exposure in shellfish, other than a razor clam, is the Dungeness crab. And as it was so well presented earlier in this webinar, the viscera, crab viscera can hold a lot of DA, a lot of domoic acid, as well as the crab meat, but the viscera holds the most. And the domoic acid, the current DA levels for being allowed to harvest crabs and crab viscera is a viscera level of 30 parts per million established by the FDA. It's a very old number. So at 30 parts per million in the viscera, you can buy Dungeness crabs. Most people remove the viscera prior to eating, but if they're boiling the crab in with the viscera, or if they're in certain cultures that eat the crab viscera or crab butter, they're at risk of higher levels of DA. I have to say the monitoring is very close, is exceptionally close in Washington, Oregon, and Northern California. But I can just say that the only other risk would be the crab, even though they don't hold it for a year. If you have an extended bloom of pseudonychia, which some of it produces domoic acid, they're going to keep getting repeatedly exposed, and so that will present a problem.
Thank you. So you had mentioned at the beginning of your talk that different species have different depuration rates, or rates at which they can get rid of the domoic acid, with crabs being on the shorter end of the scale and razor clams being on the long end of the scale. Do you have a sense about other bivalves, including mussels?
No, I think a shellfish biologist would be best to answer that. That's a great question.
Thank you. Going through our list here as more audience questions are coming through. Okay. Looks like most of these have been answered. Thank you so much, Dr. Gratton. I think we'll be moving on to our panel discussion. Next, we'll have a discussion panel led by Dr. Pam Line, Professor and Chair in the Department of Molecular Biosciences in the UC Davis School of Veterinary Medicine, a faculty member of the Mine Institute, and director of the Counteract Center for Excellence. The Line Laboratory studies cellular and molecular mechanisms by which environmental chemicals interfere with neurodevelopment and neural function to contribute to environmentally induced diseases. Our panel will feature our 4 key speakers: Dr. Tom Burbacher, Ms. Rebecca Petroff, Dr. Nina Isaharanen, and Dr. Lynn Gratton. Welcome, Dr. Line.
Thank you, Shannon. It's been a really exciting webinar. I'm really glad I was able to join in from the beginning and hear all the presentations, so thanks to all the presenters. And before we get into the actual panel discussion, I'd like to bring all of the audience back around to the mission of this webinar that was introduced to us at the very beginning, which is really to focus on the science that's come forth over the last few years, really looking at neurological and developmental effects of chronic low-level effects, or low-level exposures, I should say, to domoic acid. So with that, I'd like to lead off with the first question, which is to go round robin through all our 4 speakers and get your personal perspectives based on the science you've both done and that you're familiar with in this area, and give us your sense of what you think is really the most sensitive adverse effect that occurs at the lower doses of human exposure. So let's start with Tom.
Okay. In terms of what we know right now, you know, if I were at EPA and trying to do a risk assessment at this point in time, and sort of knowing what their risk assessment procedure includes, they typically would take the human data as the primary source, or what they usually call a critical study, and then use the animal studies to support that in terms of weight of evidence. I think that if you did do that, you would use the everyday memory outcomes as your most sensitive indicator at this point.
And do you think that the animal data supports that as one of the more sensitive adverse effects of domoic acid?
Well, we didn't do any memory assessments on our adults. We did do this observations for overt toxicity and found not overt toxicity, but a more subtle effect on motor. We think motor in terms of the tremors. The early memory scores for the infants would indicate that so far that's the most sensitive indicator in terms of infancy effects due to prenatal exposure. It's a different type of memory than I think what Lynn's talking about in terms of everyday memory. But it probably is. I would assume that it's targeting the same parts of the brain.
Thank you. Rebecca, your thoughts?
I think Tom did a very good job of explaining kind of what we would think of in the humans, but I also want to iterate that some of the studies that we did have never been conducted in humans, so it'd be important to try to follow up what we did in our non-human primate model, understand what's translatable and what's not, and then look at those results to really understand what's the most sensitive marker of toxicity in humans.
Thank you. Nina, your thoughts?
Well, I don't know that it's necessarily my area of expertise, but I do find it kind of interesting how clear-cut the tremor data was that Tom presented. I don't know if we have actually tried to assess that in humans, but I think it would be interesting to see if that happens. I agree with both Tom and Becca said until now. So.
Great, thank you. And Lynn, what do you think?
Well, obviously I thought memory would be a good in the first marker because that's where we started, and that was the most profound effect with the acute cases in Montreal, and even at postmortem they had atrophy, neuronal problems in CA1 area of the hippocampus, which has been associated with memory, and those same findings were found in sea lions and other another species, but I cannot and I would not venture to state that that's the only problem. That's just what I looked at in humans and ruled out other cognitive problems. But I can't specifically address infants, small children, or maybe special problems associated with the elderly.
Okay, thank you. So I'm going to ask a question of each of you in turn that's directly relevant to the presentation you gave. But I would encourage any of the other presenters who are part of this panel to respond, and I'll give everyone a chance to do that after the initial person I've addressed the question to has their chance to answer. So Tom, I am going to start with you. And I think the altered visual recognition in the non-human primate infants was particularly striking. I wondered if you could comment on whether you expect similar sorts of outcomes to manifest in human infants, and is there any evidence to suggest either in the non-human primate or in the human that these very early symptoms or effects in the infant will really translate into memory deficits later in development?
Good question. So the early visual recognition memory effects that we've seen are, I think I mentioned, are very similar to ones that we've seen for other toxicants. We've done studies with our animals with methylmercury exposure, in utero methylmercury exposure, and it's a widely accepted developmental neurotoxicant. We saw similar results from those studies. In developing this test, when we adapted it from humans, we also found that other high-risk groups like premature infants, low birth weight infants, sometimes also showed deficits in recognition memory. So in our model, as well as there's actually quite a body of literature, human studies, indicating that it's a fairly sensitive indicator of early memory processing, and there have been several human studies as well with other contaminants that show that there are effects on infant facial recognition memory. There's also another body of literature since this test has sort of been in active use that shows that it's a fairly good predictor of IQ later on in life. Unlike a lot of other tests that are done on human infants this young. So it's one of the hypotheses is that it's picking up very early abilities to process information effectively, which actually would be expected to correlate better with later IQ than some of the others that we have. We haven't looked again in our laboratory. It's probably been the most sensitive indicator than the other assessments that we do a little bit later in life. They tend to be not as sensitive as this one. So I think there's evidence that this is a good test in terms of picking up differences related to exposures, and it also has been shown to be a fairly good test in terms of predicting later cognitive functioning.
That's pretty interesting. Do any of the other presenters have any follow-up to Tom's response? Okay, thank you, Tom. That's actually
this
cool.
This is Lynn. Okay, I just wanted to address a couple of things. And this is Lynn Gratton. And that is that a number of the animal models, specifically mice and rats, showed some protracted problems emerging. That initially after birth in the and in the juvenile animal, there wasn't a memory problem found, but then they found it later as they got older. And secondly, more related to my study, I presented some data at the American Public Health Association meeting last year on a series of infants that our team saw in collaboration with Dr. Kim Grant from Dr. Brabacker's lab. And the data that the information that I found, now this is just a small number of cases, it was just 13 like pilot cases, showed that the kids, the babies that were exposed in utero to domoic acid, or we looked at nursing also, but it looked like it was primarily in utero, higher level in utero exposure, had delayed motor development. But they caught up by the time they were 5 years old, and there was no evidence of delays by the time they were 7 to 10 years old. So I don't know if that delay in motor development in that very handful of infants we saw would in any way be related to what Tom's seeing. But I just would like you to know we're looking at it. We'd be looking at it further, except my NIH funding was not continued. I'm disappointed, but still determined, so I will not give up answering that question.
Great, thank you. Any comments from Rebecca or Nina on the discussion thus far? Okay, so moving on then. Rebecca, I was actually really fascinated by your MRI work, really cool stuff. And so one of the things I noted is that if you think, at least the normal person who thinks about domoic acid, the non-expert in the field, the hippocampus is always the brain region that you think of being most impacted. And of course, that's a lot of the data from the acute domoic acid neurotox studies. But you're showing that there's a number of other brain regions that appear to be impacted. So these white matter tracks in the brain and other brain regions that aren't typically thought of in the context of domoic acid neurotoxicity. So I wondered if you could sort of comment on whether this provides insight into mechanisms beyond neurotoxic excitotoxicity mediated by a kainate receptor stimulation, or if it basically may give us a way to identify additional vulnerable populations.
Yeah, so there's a lot in there.
Yes, I'm sorry.
I think the first thing is kind of thinking about what I was actually able to look at with this study. So DTI only looks at white matter. The hippocampus is typically the major structure in the brain that is associated with domoic acid exposures, but it is mostly gray matter. So this particular method wouldn't actually be able to look at the hippocampus itself. That being said, the major white matter tract that does come from the hippocampus, the fornix, was impacted in our study. So it's parallel but not totally unexpected. The unexpected part was the internal capsule, which had never been looked at before. And I think this is really interesting and shows kind of what can be limiting in science sometimes, because we kind of went with a whole brain approach rather than looking at pinpointed areas, which allowed us to have this new discovery. That's not to say it hasn't been changed in other cases throughout this whole episode. I also want to note that the white monkeys is very different than the white matter tracts in humans. There are a lot of parallels in the structures, but that could certainly affect how these results translate to humans. I think the second part of your question was thinking about new vulnerable populations, correct?
Yeah, or how do your data inform what we know about susceptible populations or even mechanisms of action?
Right. So the decrease that we saw is associated with kind of a couple of different mechanisms of action, one of which could be activation of the immune cells of the brain and infiltration into the areas that we saw to be impacted. Another could be the axons of the neurons actually dying, or another one could be, and this was seen in some slice studies, so when you're actually looking at parts of the brain outside of the full animal model, could be fiber sprouting. So if axons start to generate additional fibers, that could change the measure that we saw changed in our MRI. All of these give us an idea of new areas to look at, but don't necessarily pinpoint a single mechanism. In terms of vulnerable populations, I think what's really interesting with this finding is that we can start to think about how things like co-exposures, which was kind of mentioned earlier with mercury, but we can think about other exposures in our environment, as well as other neurodegenerative diseases, can impact one's ability to withstand domoic acid toxicity. But that's about all we can do with this particular data, is think about those potential hypotheses and look to new research to really test those.
Great, thank you. Any additional follow-up from any of the other members of the panel to Rebecca's comments? Okay, thank you. So moving on, Nina, we're in an area where I don't have a lot of expertise, which is toxicokinetics. So I really enjoyed your presentation. But one of the questions that I did have for you is you seem to make the point that in the acute toxicity exposure paradigms, that really it was the peak concentration of domoic acid, which is most of concern with respect to the acute toxic outcomes. But when we're thinking about the low dose exposures, is it your feeling that it's the peak concentration that's reached, or the persistence of the exposure that's going to be more important in determining the toxic outcome?
I think that's a really, really good question. And I think one would actually have to specifically design a study. You could probably do this in rodents to see whether the peak concentrations or the duration of the concentrations drives the toxicity, because the shape of the curve is so different between the IV and oral. I completely agree with what you said, that in the acute toxicity is the peak concentrations that is driving that. But I think when we talk about the effects that, on memory or, the tremors or whatever not, I would tend to believe that it's actually the persistence of the concentrations. But one would really have to get the same AUC essentially after an IV continuous exposure versus oral continuous dosing to truly answer that question. I could design the study, I do not have the data.
Fair enough. Yeah, I think my gut feeling would also be it's the persistence rather than the peak, but you're right, until we have the data, we just don't know.
Yeah.
Another question I did have, and then I'll open up to the rest of the panel, was this idea that there might be a transporter for domoic acid is fascinating, and I think also points to another area of susceptibility, because we know there's well-known, well-described polymorphisms in the genes that encode transporters amongst humans, which could, of course, then impact disposition of domoic acid. But in terms of just the delivery from the maternal unit to the fetal unit, how is this possibility of an existence of a transporter going to impact that?
Yeah, so I think the placenta, part of our general research program that I'm part of at the School of Pharmacy is actually maternal fetal transfer or various drugs and things like that. So we've looked quite extensively in terms of metabolism versus transport in the placenta or the fetal liver. And the conclusion collectively from all the drug-related work that we've done is that the predominant protective mechanism for the fetus is placental transporters. That the placenta really has very high expression of both uptake and influx transporters, actually. So I think, I don't think the monkey placenta has been as well studied, but what we know from the human placenta is that the transporter expression actually does change as function of gestational age as well. And then the different transporters are localized at different parts of the placenta as well. So it is likely that there is a transporter that essentially pumps domoic acid from the placental cells back to the maternal unit, and that's what's limiting the exposure. Because otherwise, when we look at these babies, they had been there certainly in what we'd call a steady state. The exposure had gone on throughout the pregnancy. So that lower fetal concentration of domoic acid compared to the mom cannot be explained by not insufficient time for the domoic acid to get to the fetus, if that makes sense.
Interesting. Yeah, that's really interesting. So do you suspect, or do you know, are there transporters in the fetal blood-brain barrier that could also be influencing uptake into the fetal brain?
My understanding is that the fetal blood-brain barrier is not nearly as tight as the adult blood-brain barrier. It is not my area of expertise, though, the fetal blood-brain barrier, but what I understand, it really develops sort of across pregnancy, and it's not as tight. So one could imagine that probably in the fetus more of the domoic acid gets into the brain than in the adults.
Great, thanks. Any comments or responses from the rest of the panelists? Okay. And then my last direct question, and we'll go back to Shannon to see if there's any questions that have come in from the audience. But Lynn, really fascinating presentation that you gave. And so one of the questions would be that if you think about the coastal cohort data that you've collected in the context of the existing animal study data, what does it really tell us about dose response in the low dose range? Can you make any comments on that?
Well, my general perspective is that if you eat a whole lot, is that 20 parts per million may not be protective, or less than 20 parts per million may not be protective if people have repeated high-level exposures.
And by high level, you mean the higher end of the lower dose range.
Eating a lot of clams.
Yeah.
And I say that after also looking at the animal literature.
Mm-hmm. Okay, great. Any comments from the other panelists? I'm sorry, Lynn, did I cut you off? Did you have something else to add?
No. Thank you for asking.
Sure. So I did have one last question on my own personal curiosity, and then I will turn it over to Shannon to fill us in on what the audience is asking. But that is, you know, the tremors in both the adult female monkeys as well as in the infant monkeys is pretty interesting. And I wonder if either Tom or Rebecca would like to comment on your thoughts as to whether this is really a CNS effect or if it's really an effect at the level of the skeletal muscle system.
Well, I think both Rebecca and I are in agreement that it's a CNS effect, but if you want to.
Okay.
I think that one piece of convincing information, which I'm hoping gets published soon, is from one of our colleagues out of Woods Hole. Dr. Panlilio just finished her dissertation on domoic acid in zebrafish, and they found a lot of effects with the myelin in particular, which could be definitely associated with the tremors that we're seeing. That being said, we're certainly interested in looking at a lot of different areas that the tremors could be stemming from.
Mm-hmm. Mm-hmm. Okay, cool. All right, Shannon, do you have any questions from the audience for the panel?
Yes, and that tremor question was a great lead-in, as we have one from the audience that would like to put this to Dr. Gratton. Would it be feasible to go back and test for the tremors that were observed in the primate model in the cohort? Would it be difficult to do that? Do you still maintain contact with people that have been participating in the coastal cohort in the earlier years?
We still maintain communication with them, and I don't see any reason why we couldn't go back and look at it. I'd actually be even more interested in looking at it in aging folks in the cohort, but I think it's worth looking at in all age groups.
Thank you. And Pam, this one is for the.
Before we move on, how to say this? Assessing tremors is there's a high degree of background noise with this outcome, even in our monkeys. You probably could see that on the graphs that we have. So people tremor for a lot of different reasons, and everybody kind of tremors more as you get old. I know that for sure. So it is a difficult task to use, given the variability that you're going to get in probably your control group, sort of depending on who you're looking at. So I think we have to kind of be careful. I mean, there's also several ways to assess tremor. What we did was, as I mentioned, part of a fairly quick and easy approach just to look at what their arms look like when they're reaching for something. If you really wanted to do it in a real organized, standardized way, you can actually use accelerometers to actually quantify people's tremors, which would be, I think, a much more sensitive indicator than what we did. So yeah, so you can do it. It'll be noisy, so you'll probably need a fairly large N to be able to show differences depending on your exposures.
Yeah, I also want to point out that we're really limited with our model. It's amazing that we were able to pick up this really subtle tremor in the first place, just because we can't, unlike humans, we can't ask the monkeys to draw a spiral or touch your finger to your nose. So it's really powerful that we did see this, but again, it's going to be quite different in humans, just because, you know, essential tremor impacts so many different people and comes from so many different areas of the brain, and can be caused by disease, other toxins or toxicants, a whole host of unknown ontologies from this particular disease. Is really fascinating disease, tremoring in adults, but really unclear right now in the literature.
So we're not indicating that we should think about this. So don't be too negative.
Right.
But I mean, there are issues with it that need to be considered.
So this is Pam again. I mean, I think a lot of people, and I know tremors are not the same as seizures, but a lot of people do kind of associate chemical-induced tremors with maybe chemical-induced seizures. Is there any indication that individual animals that do exhibit tremor are more susceptible to the seizure-genic effects of domoic acid?
I wish we knew. We don't know that. As I mentioned, a few of the infants, not all the infants, had tremors. We had probably 3 or 4 that had a transient tremor outlook. As I mentioned, by about 2 months, those were pretty much gone. Part of our proposal to study them as they got older was to see whether or not those early tremors were related to any kind of motor kinds of deficits later on in life. So we were hoping to look at that and possibly even do some seizure susceptibility types of tests, but we didn't do that.
Got it. It would be a cool study, though.
Yeah.
Shannon, I turn it back to you.
So in continuation of this conversation with the human data versus the animal data, it's important to keep in mind that the messaging to the public is critical, and we must be careful with that. For example, what is the transferability of the animal model data to humans? And the questionnaire brings out the point that many folks who rely on shellfish and other seafood for subsistence dietary needs may be very impacted.
Anybody want to take a stab at that?
Well, I think that gets to the messaging in terms of how you want to interpret this data in terms of limiting any kind of seafood intake. I know that in talking with the folks from the Department of Health in Washington, and Lynn brought this up as well, that they have put out a provisional, what would you call it? Warning to limit, your consumption. So I think that that the process, my understanding of the process that they used to do that, looked at what is the current sort of acute tolerable daily intake, and put some order of magnitude on that process to bring it down to to where it was, I think, the 12 to 15 that you could eat. I think that it's probably, well, not probably, I think it would be a good idea to make sure that we, also include the message that, seafood's good for you. And because you're limiting one type of seafood doesn't mean you have to limit other types, and to make sure that, we're not changing people's diets, overall.
Yeah, and we acknowledge that the razor clam in particular is a very cultural, valuable food to a lot of populations here in the Pacific Northwest, including the Native American tribes on the coast. And I think we want to be really cognizant of that going forward and making sure that we're not really coming at this from a top-down perspective, but working with the communities to make sure that we are getting the message right and making sure everyone's protected, but also protected culturally as well.
Thank you. Anyone else on the panel have a comment? Okay, Shannon, any other questions from the audience?
Yeah, so we have a question that just came in. Are there any known immunological or endocrinological effects associated with DA exposure?
Anyone on the panel want to answer that?
There are a couple papers. They're few and far between. We did look at some hormones and some peripheral blood markers of immune function, and we did not see any differences in our non-human primate model.
So this is Pam. I know there is also literature in the sea lion indicating that at least acute domoic acid neurotoxicity can cause pretty pronounced and persistent neuroinflammation in the brain.
Thank you.
Lynn? Okay, Lynn, nothing from you? Okay, go ahead. Thanks, Shannon. Next question.
Another question comes through similar to one that we talked about earlier in the webinar. Are there ways to prepare razor clams that do degrade the domoic acid toxin? And we talked about, particularly from the standpoint of the California health agencies, that we recommend either boiling and or steaming shellfish so that, along with other types of seafood, to allow the juices to drain away so that you can discard the fluids in which you're cooking the razor clams. That DA is very heat stable and that cooking does not degrade the toxin. However, there can be a leaching effect where DA is coming out of the critter into the fluid. So we recommend that the fluid is always discarded after preparation and not used for other dishes. Great. Okay, that looks like we're up to date at the moment. Did anyone else have any? Okay.
So I guess I'll just go around the panel one more time and ask if they have any closing thoughts or statements. So we'll go in reverse order this time. So Lynn, would you like to start?
Okay, well, first of all, thank you for allowing me to participate in this important webinar. And I look forward to continuing to do research in this area and hope to keep you all posted on human health impacts as they arise. Thank you.
Thank you, Lynn. Nina?
Yes, thank you on my behalf, so including me on the panel. It's been very interesting, and guess stay tuned for the next steps.
Great, thank you. Rebecca?
Yeah, first of all, thank you to all the organizers and lovely people who put the time into organize this and dissemination of research. And I guess the thing that we really want to stress from our group over here is that given all the research and given the way that the climate is trending, we need to consider domoic acid as a public health problem that impacts not just one isolated group of people, but can impact a lot of different groups. So I want to stress that and hope that there's continued research in this arena going forward. Thank you.
Thank you, and good luck to you on your PhD research. It's a fascinating question.
Thank you. I'm hoping to finish soon.
And Tom,
she'll be looking for a postdoc if anyone's out there.
Oh, I'm looking for
So this is Tom. Yeah, I want to thank the organizers. This is done quite well, very nicely. And thanks everyone for all the attendees for listening. I guess the only thing, and it's kind of self-serving, I just wanted to remind folks if there's any research labs out there that are interested in domoic acid issues that would like to take a look at our tissue repository and do some collaborative work. Just send me an email. I hope my email's somewhere on all this information that's going out. If that's the case, then go ahead, send me an email. So thanks a lot.
Great, and I think, Shannon, that we're finished with our panel discussion.
Thank you, Dr. Lyon and panelists. A few other reminders, housekeeping items. If you are interested in seeing one of the PowerPoint presentations that were presented today, please go ahead and email me, Shannon Murphy. My address is on the screen here, and I will put you directly in touch with the authors and you can make arrangements therein. Also, too, in the control panel under handouts, we have a couple of handouts available, including today's agenda, complete with the titles of the speakers' presentations, a short bio sketch summary of all of these speakers and organizers, as well as a flyer of today's event. I will remind you as well that for those of you who are interested in the speakers and presentations that were discussed at the 2017 OEHA UC Davis workshop, we do have a copy of that agenda on our website. That might be of interest, particularly to some of you who expressed some interest in furthering discussion about marine mammals, harmful algal blooms in general. That would be a good place to start. And certainly feel free to email me if you want to be put in touch with a particular presenter from that event. I encourage everyone to visit our website at OEHA, especially our domoic acid page. We do intend to post this webinar at a future date and are exploring the technical options to do so. With that, I'd like to take the opportunity to express our great appreciation to all of our panelists, Dr. Burbacher, Ms. Petroff, Dr. Isa Hernan, Dr. Gratton, and Dr. Lyon, and to all the attendees for your contributions to today's webinar. We would also like to acknowledge our producer, Tracy Cook, OEHA scientists Becky Stanton, Wes Smith, and Tran Pham, and the OEHA IT department, especially Diane Curtis and Alina Canessa, for their critical contributions to the organizing of today's event. Tracy, our producer, do you have any further announcements before we make our closing remarks?
I don't, but thank you. I thought the session today was great.
Thank you so much for all of your work behind the scenes. Thank you, Tracy. Now here is our OEHA director, Lauren Zeiss, with some final thoughts.
Well, my final thoughts is just a big thank you all for participating on the webinar, sending in your questions, and thank you to the panelists and the speakers and Pam Lyon for such a masterful job in the last session. And I just want to close the meeting and thank our OEHA staff very much for a job very well done. So thank you all.
Thank you, everyone. Goodbye.
Thank you.
Bye-bye.